In the stillness of the evening, when light fades gently into darkness and the body begins to soften into rest, there exists a quieter world beneath the surface of your skin, a world of motion, exchange, and gentle conversation. Though imperceptible to the eye or ear, this world is always alive, always communicating. Inside you are trillions of cells, each one a self-contained unit of life, quietly performing its own tasks with remarkable precision. A red blood cell drifts through your bloodstream, ferrying oxygen to waiting tissues. A liver cell processes nutrients with practiced efficiency. A neuron prepares to pass along its next electric impulse. Bone cells strengthen your frame. Skin cells create your outer shield. Immune cells roam in silent vigilance. Despite their differences, none of these cells exists in isolation. Though small, many smaller than a grain of dust, they do not act independently or alone. They are collaborators in a grand biological orchestra, playing in synchrony without conductor or score. What unites them is not hierarchy or control, but something more subtle: communication. This cellular communication is quiet but constant. It is the heartbeat behind every function of life, from the mundane to the extraordinary. It ensures your body knows when to sleep and when to wake, when to heal a wound, when to grow, and when to conserve its resources. Even as you lie still, your cells are busy speaking to one another, exchanging messages in a language of molecules, ions, and electrical pulses. To communicate, cells use a variety of methods, depending on their location and purpose. Some reach out physically, touching their neighbors through small protein bridges that link cell to cell. Others send chemical messengers, hormones, neurotransmitters, cytokines, drifting like tiny messages in a bottle through the blood or the fluid between tissues. Still others rely on the silent flicker of electrical charges, pulsing through nerve cells to carry urgent signals from brain to limb and back again. Each of these methods is precise. Each has its own rhythm and destination, and each plays a role in maintaining the body's delicate internal balance, a state known as homeostasis. This balance is not static, but dynamic. Imagine a lake on a calm day, its surface still, its depths quietly teeming with life. Beneath that apparent stillness, temperature changes, light penetrates in shifting layers, nutrients rise and fall. In the same way, your body is constantly adjusting itself, moment by moment, in response to internal signals and the world outside. Perhaps your blood sugar dips slightly. A signal is sent. Hormones are released. The liver responds, converting stored molecules into usable energy. Or perhaps a virus is detected. Immune cells are summoned through molecular distress signals, each one guided by the language of cell-to-cell messaging. These messages do not shout. They do not panic. They flow gently, reliably, ensuring the body responds with care, not chaos. Even when you are unaware, your cells are listening and responding. In the darkened interior of your brain, neural networks are exchanging signals that regulate heart rate and breathing. In your gut, sensory cells monitor digestion, while others keep an eye on hydration and nutrient absorption. In your skin, tiny receptors respond to changes in pressure, temperature, and even the brush of fabric against your arm. All of this is communication, not with sound or light or voice, but with molecules shaped like keys, designed to fit only the right locks, with small shifts in electrical charge, carefully controlled, with pathways so ancient they evolved long before mammals, birds, or even fish. This is not the kind of communication we associate with language, but it is a language nonetheless. It is the grammar of life, written not in words, but in chemicals and currents. And it is so finely tuned that a single misfired message can have consequences. A signal sent too often, or not at all, can lead to imbalance, to disease, fatigue, confusion in the body's system. That too is why cells communicate, not just to do their work, but to ensure their work is done in coordination with others. The story of cellular communication is one of cooperation. No cell, no matter how specialized or important, functions entirely on its own. A heart cell contracts only because a signal tells it to. A lung cell opens to receive oxygen only because it was prompted. And every signal is both an answer and an invitation. "This is what I am doing," the cell says. "What should you do in return?" And so, the entire organism, your body, becomes a place of silent cooperation, a place where information flows steadily in all directions, upward to the brain, outward to the skin, inward to the organs, across the tiniest synapse between neurons. Every breath, every blink, every beat of your heart is shaped and guided by this flow. It is easy to forget, perhaps, that this is happening. Easy to imagine the body as a solid, silent form. But in truth, you are more fluid than fixed, more network than machine. Every cell inside you, every one of the trillions, is busy both speaking and listening, and doing so in rhythms that have persisted for billions of years. Long before there were humans, before eyes or hands or voices, cells were communicating. Life, in its earliest forms, relied on this capacity to send and receive signals. It is one of the oldest characteristics of life itself. Tonight, as you rest, this communication continues without your awareness. As your body settles into stillness, your cells do not sleep. They carry on their quiet tasks, guided by the unending exchange of information. And just like the moonlit surface of a quiet lake, their activity reflects a balance deeper than it first appears, a balance maintained not by chance, but by careful, continuous conversation. In the chapters that follow, we will explore the many ways this conversation unfolds. We will examine how cells speak, how they listen, and what happens when their messages fall silent or go unheard. We will move gently through the molecular signals, the pathways, the gates and the pulses, not as test subjects or scientists, but as quiet observers, watching the hidden harmony within us. But for now, let this thought accompany you into deeper rest, that within you, even now, a peaceful symphony of whispers plays on messages passed from cell to cell, ensuring that the body remains whole, steady, and at peace. 2. The purpose of communication in living systems. The ability of cells to communicate is not simply a matter of convenience. It is not an optional feature of life, something that could be set aside without consequence. It is, instead, a fundamental necessity. Without it, life as we know it could not exist. When we think about communication in the human sense, we might imagine speaking, writing, sending messages across distances. In the realm of living systems, the concept is both more ancient and more essential. For a cell, communication is how it survives, how it cooperates, and how it contributes to the greater whole of the organism it belongs to. It is how the body functions not as a random collection of parts, but as an integrated, harmonious entity. Cells are, in a way, specialists. Each has its own role, its own duties to perform. Muscle cells contract to produce movement. Red blood cells ferry oxygen to every corner of the body. Immune cells patrol for signs of infection. Neurons carry signals that coordinate everything from reflexes to thought. These roles are distinct, yet each depends on the others. A muscle cell cannot create oxygen for itself. It relies on the circulatory system to bring it. A neuron cannot fight an invading bacterium. It depends on immune cells to respond. This interdependence means that cells must constantly be aware of the state of their neighbors and of the organism as a whole. They must know when resources are plentiful and when they are scarce. They must recognize signals of danger and signals of safety. They must sense when growth is needed and when it would be wasteful. Consider the simple act of healing a small cut on your skin. The injury begins with a disruption. Cells at the site are damaged, and the protective barrier of the skin is broken. Immediately, these damaged cells send out chemical distress signals. Nearby blood vessels respond by widening, allowing immune cells to pass through and reach the site more easily. Platelets, tiny cell fragments, gather to form a clot, while fibroblasts move in to begin rebuilding the tissue. All of this is coordinated through an intricate web of communication. Without it, the wound would remain open and vulnerable. The same is true for growth and development. From the earliest stages of life, cells send and receive signals that determine their fate, whether they will become part of the skin, the muscles, the bones, or the nervous system. These developmental cues must be precise. If they go awry, the body's form and function could be compromised. In this way, communication ensures that each cell knows not just how to perform its tasks, but also when and where to do so. Even in a body at rest, communication is vital for maintaining homeostasis, that delicate internal balance that keeps us alive. Imagine the temperature of your body rising just a little too high. Cells in the brain's hypothalamus detect this change and send signals that increase blood flow to the skin, activating sweat glands to release cooling moisture. If the temperature dips too low, the same system sends signals to constrict blood vessels near the skin, conserving heat, and to activate shivering in the muscles to generate warmth. Similar balancing acts occur with water levels, blood pressure, pH, and nutrient availability. Cells exchange information about these conditions constantly, adjusting their own activity in response to the greater needs of the body. It is an unending negotiation, a consensus reached not by command, but by conversation. In multicellular organisms, the purpose of communication extends beyond individual survival. It supports the survival of the whole. A single liver cell, for instance, may never meet a skin cell directly, but its work in filtering blood affects the health of every skin cell in the body. The reverse is also true. The skin's ability to keep out harmful substances protects the liver from being overwhelmed. This mutual benefit is only possible because each cell plays its part in response to information received from others. Communication also allows for swift and coordinated responses to the environment. In animals, the nervous system uses electrical and chemical signals to alert the body to changes: a sudden drop in temperature, a new source of food, a potential threat. The endocrine system complements this with longer-lasting hormonal signals that adjust metabolism, growth, and energy use. Together, these systems ensure that the organism can adapt smoothly, without chaos or confusion. Even in the smallest living beings, single-celled organisms like bacteria, communication serves a clear purpose. Bacteria can release chemical signals into their surroundings, allowing them to sense how many of their kind are nearby. When their numbers reach a certain threshold, they can change their behavior collectively, perhaps forming a protective film or releasing a burst of enzymes to digest available food. This process, known as quorum sensing, is a reminder that the need for coordination is as old as life itself. It is worth noting that communication is not only about action. Sometimes, the most important message a cell can send is, "All is well." In times of stability, these reassuring signals help maintain calm and prevent unnecessary changes. The absence of such messages, however, can be a warning, a sign that something has gone wrong. In cancer, for example, cells may stop listening to signals that tell them to stop dividing, leading to uncontrolled growth. In autoimmune conditions, the immune system may misinterpret signals from healthy cells as signs of danger, launching an attack where none is needed. The purpose of cellular communication, then, is threefold: to coordinate activity, to maintain balance, and to adapt to change. These purposes are deeply interwoven. Without coordination, balance is lost. Without balance, adaptation becomes frantic and wasteful, and without adaptation, survival is threatened. One might picture this in terms of a symphony orchestra. Each section, strings, woodwinds, brass, percussion, plays its part. Communication ensures they all keep the same tempo, adjust to the same key, and follow the same progression. If one section rushes ahead or falls behind, the music suffers. In the body, cells keep time with one another not through sight or sound, but through a ceaseless flow of molecular and electrical signals. This is why communication is more than a background process. It is the essence of life's organization. It transforms a collection of cells into a unified organism. It allows your body to heal, to grow, to respond to the world, and to remain steady in the face of constant change. As you rest tonight, this purpose continues to be fulfilled. Cells deep within your muscles monitor their own needs and adjust energy use. Cells in your blood vessels fine-tune pressure and flow. Cells in your brain quietly exchange signals to maintain the rhythm of your heartbeat and the depth of your breath. You are not aware of it, yet it is happening in every moment, a testament to the quiet, unbroken purpose of communication in living systems. 3. The microscopic language of life. Every form of life, from the smallest bacterium to the tallest tree, depends on communication. But for cells, there is no spoken word, no written letter, no sound carried on the air. Their conversations are quieter, their language invisible to human eyes. And yet, in its own way, it is every bit as rich as any language we speak. Cellular language is built from a set of distinct dialects. Each has its own medium, its own style, its own range of expression. Together, these dialects allow cells to share an astonishing variety of information: when to grow, when to rest, when to change, and when to act. Just as human language can convey facts, emotions, and subtle shades of meaning, the languages of cells can express urgency, reassurance, cooperation, or alarm. One of the simplest and oldest of these languages is direct contact. Imagine two neighbors leaning over a shared fence to exchange a few words. In the cellular world, this is done through physical structures that join cells together. In animals, tiny protein channels called gap junctions link the interiors of neighboring cells, allowing small molecules and ions to pass directly from one to the other. In plants, similar structures called plasmodesmata connect cells across their sturdy cell walls. Through these microscopic doorways, cells can send messages instantly, without the delay of sending them through surrounding fluid. This is the language of immediacy, direct, personal, and unambiguous. Then there is the language of chemical messengers, perhaps the most widely used dialect in all of biology. This is the language of hormones, neurotransmitters, and growth factors, molecules released by one cell and received by another. Like letters sent through the post, these messages travel from sender to recipient, each bearing information in its unique molecular shape. The words of this language are chemicals designed to fit perfectly into the locks of specific receptors on the receiving cell's surface or within its interior. Some of these messages travel only short distances. In paracrine signaling, cells send out molecules meant for their immediate neighbors. These short-range messages are like quick notes passed between desks, useful for coordinating activity in a small area. Other messages are meant for long-distance travel. In endocrine signaling, cells release hormones into the bloodstream, allowing them to reach targets far away, sometimes on the other side of the body. These are the long letters, sent across vast distances, that still arrive precisely where they are needed. Another dialect is the rapid, precise language of electrical signals. In nerve cells, or neurons, the movement of charged particles, ions, across the cell membrane creates tiny electrical currents. These currents travel down the length of the cell, carrying information with remarkable speed. When they reach the end of the neuron, they trigger the release of neurotransmitters, bridging the gap to the next cell. This is the language of urgency, capable of moving messages from your toes to your brain in the blink of an eye. Cells can also combine these languages, shifting fluidly from one to another as the situation demands. A neuron may send an electrical impulse along its length, then translate it into a chemical message at its tip. An immune cell might respond to a chemical signal by physically binding to another cell, delivering information through direct contact. These blends of communication create a subtlety and flexibility that ensures the message is not just sent, but fully understood. The microscopic language of life is not limited to signaling between cells of the same type. Sometimes messages must cross the boundaries between very different kinds of cells. In your body, for example, nerve cells must communicate with muscle cells to coordinate movement. The language they share is a specialized form of chemical signaling, designed so that the muscle cell responds instantly to the neuron's instruction. In plants, cells in the roots must signal to cells in the leaves when water is scarce. This message may be carried in the flow of plant hormones, prompting the leaves to conserve moisture. Even more remarkable is the ability of some messages to pass between entirely different organisms. A flower releases chemical scents that attract bees. The bees' sensory cells detect these molecules and translate them into a behavioral response to land, to gather nectar, to pollinate. In the soil, plant roots release compounds that influence the growth of beneficial fungi, and in return, the fungi signal back, initiating a partnership that benefits them both. No matter the form, all of these languages share a fundamental principle. The message must be recognized by the recipient. Without recognition, the signal is meaningless, like a letter delivered to a mailbox without a matching address. This is where receptors come into play. These specialized proteins, embedded in the cell membrane or floating within the cell, are the ears and eyes of the cell. They read incoming signals by binding to them, triggering changes inside the cell that lead to a response. The diversity of signals is matched by the diversity of responses they can trigger. A single hormone might cause one type of cell to divide, another to produce a protein, and a third to stop its activity altogether, all because each cell interprets the message in its own way, according to its role and condition. This is why the same chemical can have very different effects in different tissues. It is as if the same sentence, spoken to different people, could inspire laughter, reflection, or action, depending on who hears it. It is important to remember that the microscopic language of life is not static. It evolves, adapts, and changes over time. Cells can become more sensitive to certain messages by increasing the number of receptors they have, or they can become less sensitive by reducing them. This ability to adjust their listening is a form of fine-tuning, ensuring they respond appropriately to the conditions around them. In times of stress or injury, cells may speak more urgently, releasing large amounts of signaling molecules to rally their neighbors. In times of calm, they may speak more softly, sending only the signals needed to maintain steady function. Like any living language, the tone, frequency, and intensity of cellular communication change with the situation. If you could somehow stand inside this invisible conversation, you would hear no sound, yet you would sense a constant pulse of activity molecules drifting between cells, electrical ripples running along slender fibers, proteins fitting neatly into their matching shapes, like keys and locks. You might see colors shifting as ions move, as receptors change shape, as enzymes set off cascades of chemical reactions inside the cell. And if you stayed long enough, you would realize that this is not random chatter. It is purposeful, precise, and deeply coordinated. It is the language of life itself, the unbroken conversation that keeps each organism whole, responsive, and alive. In the coming sections, we will slow down and explore each of these languages in more detail. We will see how direct contact lets cells whisper to their closest neighbors, how chemical messengers travel like carefully addressed letters, and how electrical impulses race along nerve fibers to deliver urgent news. We will look closely at how messages are sent, how they are received, and how they are translated into action. For now, it is enough to know that beneath the surface of your skin, this language is being spoken continuously. It never falls silent. Even as you rest, it is shaping your heartbeat, your breath, your digestion, your healing, all without you ever needing to speak a single word. 4. Modes of cellular signaling. In the intricate world of cellular life, communication is not a single, uniform process. Just as people have many ways to speak with one another, a conversation across a table, a letter sent by post, a quick phone call, or a subtle gesture, cells too have a variety of modes for sending and receiving their messages. These modes differ in speed, distance, intimacy, and purpose. Understanding them is like being introduced to the different channels of a quiet, invisible network. Each is suited to a particular kind of message. Some are for urgent instructions, some for long-term coordination, and some for intimate exchanges between cells that share a direct boundary. While each mode can operate on its own, in living systems they often overlap, reinforcing and refining one another. The most immediate form of communication occurs when cells are physically connected to one another. In animals, tiny protein channels called gap junctions form a direct bridge for ions and small molecules to pass from one cell to the next. In plants, similar structures known as plasmodesmata thread through the sturdy cell walls to connect the interiors of adjacent cells. This form of communication is intimate and precise, requiring cells to be side by side, touching at their edges. Messages travel instantly through these microscopic doorways, unfiltered and without delay. It is like leaning close to a trusted friend and whispering in their ear, an essential connection in tissues where timing must be exact, such as in heart muscle cells beating in unison, or in plant cells directing the flow of nutrients between roots and leaves. Some messages do not require such closeness. In paracrine signaling, a cell releases molecules into the surrounding fluid, where they drift a short distance to affect nearby cells. This mode works locally, influencing the behavior of cells in the immediate neighborhood. It is a way for small groups of cells to coordinate their actions without involving the entire organism. During wound healing, for example, cells near the injury release growth factors that prompt surrounding cells to divide and migrate toward the damaged site. These signals travel just far enough to rally those who can help, and because they are quickly degraded or absorbed, their effects are temporary, ensuring that the message stays local and specific. Other signals are meant to travel far across the body. Endocrine signaling is the mode used when a cell releases hormones into the bloodstream. These chemical messengers are carried to distant targets, sometimes on the opposite side of the body. It is the long-distance postal service of the cellular world. While endocrine messages are slower than electrical signals, they have the advantage of lasting longer and influencing many tissues at once. Hormones from the thyroid gland, for example, regulate the metabolic pace in nearly every cell. The adrenal glands release cortisol during times of stress, preparing multiple systems for sustained alertness and adaptation. These hormones can reach almost any part of the body through the bloodstream, yet only cells with the right receptors, the correct address, will recognize and respond to the message. The nervous system, too, has its own specialized form of communication. In synaptic signaling, neurons transmit electrical impulses along their length, carrying information rapidly from one point to another. When this electrical signal reaches the end of the neuron, it is converted into a chemical message as neurotransmitters are released across the tiny gap of the synapse. These molecules bind to receptors on the receiving cell, often another neuron, but sometimes a muscle or gland cell, and trigger a rapid, precise response. This is the language of urgency, capable of moving instructions from your brain to your fingertips in less than the blink of an eye. It is the quick, clear whisper that allows thoughts to become movement and sensations to become awareness. There are times when a cell speaks not to its neighbors or distant partners, but to itself. In autocrine signaling, the cell releases a molecule that binds to receptors on its own surface. This self-directed communication can help fine-tune its own behavior, reinforcing a decision it has made or adjusting its sensitivity to other incoming messages. This is especially important in immune responses, where a cell that has detected a threat may stimulate itself to increase its defensive output. It also plays a role in growth regulation, helping a cell decide whether to divide, rest, or change its function. Electrical signals are not the exclusive domain of neurons. In heart muscle, for example, direct electrical coupling through gap junctions ensures that all cells contract together, producing the steady rhythm that sustains life. In some plants, electrical impulses travel from cell to trigger responses to touch or injury. These signals can be fast yet broad, affecting many cells in a tissue at once, more like a ripple spreading across a still pond than the tightly focused transmission of a nerve fiber. In the living body, these modes rarely work in isolation. A single event often triggers several forms of communication at once, each reinforcing and guiding the others. Consider a moment of sudden stress. Sensory neurons detect a potential threat and send electrical signals to the brain through synaptic pathways. The brain responds by directing nerve impulses to muscles, preparing them for rapid action, and by activating endocrine glands to release hormones that sustain alertness. Meanwhile, cells in local tissues may engage in paracrine and autocrine signaling to adjust their own readiness, shifting metabolism, blood flow, and immune activity. The result is a seamless, coordinated response that involves short-range whispers, long-range letters, and self-directed reminders, all working together without pause. It is tempting to think of these modes as separate languages, each with its own vocabulary and rules. But in truth, they are more like different voices within a single conversation. Direct contact is the quiet murmur between close companions. Paracrine messages are the words shared among neighbors. Endocrine hormones are the letters sent to distant friends. Synaptic impulses are the urgent calls that must be answered immediately. Autocrine signals are the private reflections that help a cell know itself. Each voice has its purpose, and together they weave the unbroken dialogue that keeps the organism alive and in balance. In the end, modes of cellular signaling are not just scientific classifications. They are the ways in which life organizes itself, moment by moment. They make it possible for every part of a living system to remain aware of every other part, to adjust to change, to heal, to grow, and to respond. As we move forward, we will look more closely at these modes in action, seeing not only their structure and mechanics, but also the quiet beauty of the roles they play in sustaining the harmony of life. 5. Communication by direct contact. Among the many ways cells communicate, perhaps the most intimate is the quiet conversation that happens when two cells are physically connected. It is a form of communication without distance, without delay, a message passed directly from one to the other through a shared microscopic link. In this closeness, information flows instantly, a seamless exchange that keeps whole communities of cells in perfect harmony. In animals, one of the most remarkable examples of this connection is the gap junction. Picture two neighboring cells, their outer membranes pressed gently together. At the point where they meet, special proteins called connexins form a narrow channel that links the fluid interior of one cell to the fluid interior of the other. These channels are so small that only ions and tiny molecules can pass through, yet this is enough to carry essential information. Through gap junctions, cells share not only nutrients but also electrical impulses and chemical signals that keep their activities synchronized. In heart muscle, this direct passageway is vital. The heartbeat is not just the result of individual cells contracting on their own schedule. It is the coordinated contraction of millions of muscle cells, each pulling in unison. Electrical signals spread rapidly from one cell to the next through gap junctions, ensuring that the entire heart contracts in a steady, unified rhythm. Without these connections, the heartbeat would become disordered, and the life-sustaining pump of the heart would falter. Gap junctions also play a role in the brain, where certain groups of neurons communicate directly with each other in this way. While much of the nervous system uses chemical signals across synapses, direct electrical coupling through gap junctions allows for faster coordination in specific networks, particularly those involved in rhythmic activity like breathing. In plants, the equivalent structures are known as plasmodesmata. Plant cells are enclosed within strong cell walls. Yet even here, nature has found a way to bridge the gap. Plasmodesmata are tiny tunnels that pass through these walls, lined with membrane and containing strands of cytoplasm that directly connect one cell's interior to another's. Through these delicate passageways, plants move not just small molecules but also proteins, RNA, and other important information. In a sense, the cells of a plant are all part of one continuous, interconnected network. This connectedness is essential for plant life. Through plasmodesmata, cells deep within the root can send signals upward, perhaps warning that water is scarce. Leaves, in turn, can send messages downward, instructing roots to adjust their growth or absorb more nutrients. This long-distance conversation, cell to cell, is what allows a plant to respond as one organism to changes in light, temperature, and moisture. Direct contact is not only about sharing through channels. Sometimes, cells communicate by recognizing and binding to specific proteins on each other's surfaces. When two such matching molecules meet, they fit together like pieces of a puzzle, triggering changes inside both cells. This is how immune cells, for example, identify and respond to harmful invaders. An immune cell will physically attach to another cell, reading the molecular identification tags on its surface. If the pattern is recognized as friendly, the immune cell will pass by. If it is foreign, the immune cell may release a series of signals that set off a defensive response. These face-to-face meetings between cells are precise, deliberate encounters. They are not casual touches, but carefully regulated interactions that occur only when the right conditions are met. This ensures that important messages are not sent or received by mistake. One of the most important qualities of direct contact communication is its immediacy. Because the message passes from one cell to another without traveling through surrounding fluids, there is no delay in its delivery. It is also a private form of communication. The message is exchanged only between the cells that are physically connected. In this way, it is very different from chemical messages released into the bloodstream, which can travel far and potentially influence many cells. The intimacy of this form of signaling also means it is deeply tied to trust and coordination. A cell opening a gap junction to its neighbor is, in a sense, opening its inner world. It allows not just messages but also part of its own chemical environment to flow across the boundary. This closeness requires careful control. Cells can open and close these channels depending on the circumstances. In times of injury or infection, they may close the channels to protect themselves and prevent harmful substances from spreading. In times of growth and repair, they may open them wide to share resources and coordinate activity. Direct contact signaling is, in many ways, the most ancient form of communication in living systems. Long before the development of complex hormones, long before the nervous system existed, cells were touching and sharing through these intimate connections. It is easy to imagine the earliest communities of cells, perhaps simple clusters, floating in ancient seas, linked together by tiny openings that allowed them to act as one. That same principle, refined over billions of years, still operates in your own body today. Even as you rest, direct contact communication continues its quiet work. In your heart, Gap junctions pass the wave of each heartbeat from cell to cell. In your skin, cells pass nutrients and ions to their neighbors to keep the outer barrier strong. In your brain, certain neurons link together in electrical unity to keep breathing smooth and regular through the night. And if you were a tree or a flower, plasmodesmata in your cells would be busy carrying information from root to leaf, ensuring that every part of you knew what the others were experiencing. It is a reminder that, at the smallest scale, life is a community. No cell exists entirely alone. Through direct contact, cells remain aware of one another, sharing not just information but a kind of unity, a sense of being part of something larger than themselves. The messages they pass through these tiny bridges are the threads that hold the fabric of living tissue together. When we think of communication, it is easy to imagine words carried through the air or signals sent over great distances. But sometimes, the most powerful messages are the ones delivered face to face, directly, without anything in between. In the world of cells, that closeness can mean the difference between chaos and coordination, between disorder and harmony, between life and the steady, quiet rhythm that sustains it. 6. Local chemical messaging. Not all cellular conversations happen face to face. Sometimes a cell needs to speak to those nearby without touching them directly, sending a message that drifts softly through the surrounding space until it reaches the right listener. This form of communication is known as local chemical messaging, and in biology it is often called paracrine signaling. It is a way for cells to coordinate their behavior within a neighborhood, ensuring that the needs of the local environment are met with gentle precision. Imagine a small community where people share information by leaving notes on doorsteps. The message does not travel far only to the houses on the same street, but it carries important details about shared needs and common plans. In the same way, local chemical messaging allows groups of cells to pass along short-range instructions that guide their work together. The signals are released into the fluid between cells, where they can be picked up by others in the immediate area. These messages are made of molecules, often proteins, small peptides, or other chemical compounds carefully shaped to be recognized by specific receptors on the receiving cell. Once a molecule binds to its receptor, it sets off a cascade of changes inside the cell, leading to the desired response. Because these molecules are quickly broken down or absorbed, their effects are temporary. This ensures the message remains local and does not spread beyond the intended audience. One of the most familiar examples of paracrine signaling happens during wound healing. When skin is damaged, the cells at the injury site release growth factors into the surrounding tissue. These growth factors act as signals to neighboring cells, telling them to divide, migrate toward the wound, and help rebuild the missing tissue. The signals travel only a short distance and fade quickly, ensuring that the repair effort is concentrated where it is needed, and not elsewhere in the body. Another example occurs in the immune system. When a pathogen enters the body, immune cells stationed in the affected area release chemical messengers called cytokines. These molecules alert nearby immune cells to the presence of a threat, prompting them to increase their activity and prepare for defense. Again, the reach of this message is limited. It is a rallying call, meant for local defenders, not for the entire immune army at once. In the developing embryo, paracrine signaling helps guide the formation of tissues and organs. Certain groups of cells release morphogens, molecules that diffuse through the surrounding tissue and influence the fate of nearby cells. Depending on the concentration of the morphogen a cell receives, it may develop into one type of tissue or another. This process requires precise control, as even small changes in signal strength or range can alter the delicate patterning that shapes the growing organism. Local chemical messaging can also be used to share resources and coordinate metabolic activities. In some tissues, cells release signals that encourage their neighbors to increase or decrease the uptake of nutrients depending on availability. This prevents one group of cells from over-consuming while others go without. It is a form of quiet cooperation that ensures the stability of the whole community. One of the important features of paracrine signaling is its ability to create gradients of information. Because the molecules spread out from the point of release and weaken as they move away, cells at different distances receive different amounts of the signal. This allows for fine-tuned responses based on position. A cell close to the source may respond strongly, while one farther away may react more subtly, or not at all. In this way, paracrine signals can create patterns of activity across a tissue, guiding development, repair, and adaptation. The short-lived nature of these messages is another safeguard built into the system. Because the molecules are broken down quickly by enzymes or absorbed by cells, the signal fades naturally once the task is complete. This prevents unnecessary or prolonged changes that could disrupt the balance of the tissue. It also allows the system to reset, ready to send new messages as conditions change. Local chemical messaging is not limited to humans or animals. Plants also use this form of signaling to coordinate growth and respond to their environment. Cells in a growing shoot may release hormones that influence the activity of nearby cells, guiding the shape and direction of growth. When a plant is attacked by an insect, damaged cells can release chemicals into the surrounding tissue that prompt nearby cells to produce defensive compounds, strengthening the plant's resistance in the immediate area. Even single-celled organisms make use of short-range chemical communication. Bacteria, for instance, release small signaling molecules into their environment as part of a process known as quorum sensing. By detecting the concentration of these molecules, each bacterium can gauge how many others are nearby. When enough are present, the group can coordinate its behavior, perhaps forming a protective biofilm or producing a shared enzyme to break down food. While the term paracrine is not usually applied to bacteria, the principle is the same. Local signals guide the actions of a community. If we were to shrink ourselves down and stand among the cells during such an exchange, we might see molecules drifting gently between them, like pollen carried on a faint breeze. Some molecules would be caught almost immediately by nearby cells, triggering swift reactions. Others would drift a little farther before being captured, and a few would fade away, their message dissolved before it could be heard. The scene would be dynamic yet peaceful, a constant ebb and flow of signals that keep the local community in balance. In the living body, paracrine signaling often works alongside other forms of communication. The same group of cells might also share information through direct contact with immediate neighbors or send long-distance instructions through hormones in the bloodstream. But the beauty of local chemical messaging lies in its specificity. It focuses attention exactly where it is needed, without disturbing parts of the body that have no role in the task at hand. This precision makes paracrine signaling a cornerstone of coordination in multicellular life. It allows for rapid, adaptable responses to local conditions while preserving the overall stability of the organism. It is the voice of the neighborhood, the soft but important conversation that keeps each small part of the body in step with the others nearby. Even now, as you read and breathe, local chemical messages are being exchanged in countless parts of your body. Cells in your skin adjust their activity in response to signals from their neighbors. Immune cells stationed in your lungs send quiet reassurances to one another that all is well. In your muscles, local signals help coordinate the energy needs of different fibers, ensuring that work is shared and balanced. These are the everyday exchanges that sustain life without ever troubling your awareness. When we think about communication within the body, it is easy to focus on the dramatic, the rush of hormones, in a moment of stress, the quick spark of a nerve impulse. But beneath these grand gestures lies the gentle hum of local messaging, a constant, unassuming exchange that makes all the rest possible. It is the quiet voice in the crowd, speaking only to those close enough to hear, yet shaping the way the whole community moves and works together. 7. Hormonal communication across distances. Some messages in the body are meant to travel much farther than the immediate neighborhood of cells. They are not local whispers, but carefully written letters, carried across vast internal landscapes to reach their intended recipients. This is the work of hormones, chemical messengers that move through the bloodstream, connecting distant parts of the body in a slow but enduring conversation. Hormonal communication belongs to the endocrine system, a network of glands and tissues dedicated to producing and releasing these signals. Once a hormone enters the bloodstream, it has the potential to reach nearly every cell in the body, yet it does not speak to all of them. Only cells with the correct receptors, the proper mailbox, can receive and interpret the message. This selectivity allows hormones to act with precision, despite their wide distribution. The beauty of hormonal communication lies in its ability to coordinate complex processes that involve many organs and tissues at once. Growth is one such process. The pituitary gland, a small structure at the base of the brain, releases growth hormone into the blood, influencing bone, muscle, and connective tissue throughout the body. This single messenger helps regulate the pace and pattern of physical development from childhood through adulthood. Metabolism, too, is guided by hormonal signals. The thyroid gland, shaped like a butterfly at the front of the neck, secretes hormones that set the body's metabolic tempo. When these hormones are abundant, cells burn energy more quickly, producing heat and supporting an active state. When levels are lower, the body shifts into a slower rhythm, conserving resources. This fine-tuning ensures that energy production matches the body's needs, whether at rest or in motion. In times of stress, hormonal communication takes on a protective role. The adrenal glands, perched atop the kidneys, release cortisol and adrenaline into the bloodstream. Adrenaline works quickly, increasing heart rate, sharpening focus, and directing blood to the muscles. Cortisol's effects are longer lasting, helping the body maintain alertness and supply energy over extended periods. Together, they prepare the body to respond to challenges, sustaining both immediate action and endurance. Hormones also govern reproduction. In women, the ovaries produce estrogen and progesterone, which regulate the menstrual cycle, support pregnancy, and influence many other aspects of health. In men, the testes produce testosterone, guiding the development of reproductive tissues and contributing to muscle growth and bone density. These hormones travel far from their point of origin, affecting systems and tissues across the entire body. The pace of hormonal communication is gentle compared to the speed of nerve impulses. It can take seconds to minutes for a hormone to reach its target and begin producing noticeable effects. But the influence of these signals is often lasting. While a nerve impulse might prompt a muscle to twitch and then fade instantly, a hormonal signal can sustain a physiological state for hours, days, or even longer. This makes hormonal communication well suited to guiding long-term processes like growth, reproduction, and metabolic balance. Because hormones circulate widely, they also have the ability to integrate information from different parts of the body. For example, the pancreas monitors blood sugar levels and releases insulin when glucose is high. Insulin travels through the blood to many tissues, prompting cells to absorb glucose for energy or storage. This single signal coordinates the work of the liver, muscles, and fat cells, keeping blood sugar within a healthy range. Without such coordination, energy supply would become chaotic, swinging unpredictably between surplus and shortage. The journey of a hormone is not random. Although the bloodstream flows everywhere, the distribution of receptors determines where the signal is received. A hormone molecule might pass thousands of cells without effect, then bind instantly to one with the correct receptor, unlocking a cascade of changes inside. This specificity prevents confusion and ensures that each hormone's message is delivered only to those who are meant to hear it. There is also a quiet elegance in the way hormones are regulated. Feedback loops maintain balance, preventing signals from becoming too strong or too weak. If a hormone's effects are excessive, the body can reduce its production or increase its breakdown. If levels are too low, production can be increased until balance is restored. This constant adjustment keeps the endocrine system responsive yet stable, much like a thermostat that maintains a comfortable temperature. Hormonal communication does not exist only in humans. Across the natural world, animals and plants use long-distance chemical messengers to coordinate their growth and behavior. In plants, hormones like auxins and gibberellins travel from one region to another, guiding stem elongation, fruit ripening, and flowering. In animals as small as insects, hormonal signals regulate metamorphosis, reproduction, and seasonal behavior. The principle is the same. A molecule is sent far from its point of origin to influence cells and tissues that are not directly connected to the sender. If we could observe this process within the body, we might picture it as a river system. Hormones are like carefully sealed messages dropped into the current, drifting along until they reach the right shore. The river touches many places, but only certain doors are open to receive what the message contains. Once inside, the signal changes the course of the cell's work, aligning it with the needs of the whole organism. The effects of hormonal communication are not always dramatic in the moment. Often they are subtle, shaping the body's function quietly over time. The steady release of thyroid hormone maintains the body's warmth through a cold night. The gentle ebb and flow of reproductive hormones influence mood, energy, and cycles of fertility. Even in sleep, growth hormone is released in pulses, supporting tissue repair and renewal without disturbing the body's rest. Hormonal messages can also influence one another. A change in one hormone's level may trigger adjustments in others, creating an intricate web of interactions. This network allows the body to respond in a coordinated way to complex situations. In times of fasting, for instance, insulin levels fall while other hormones rise to encourage the release of stored energy. In preparation for physical activity, adrenaline may work alongside glucagon to ensure that muscles have the fuel they need. The strength of hormonal communication lies in its reach and its endurance. It binds together the farthest points of the body, allowing them to act as one. It is the reason your stomach can prepare for a meal before food arrives, the reason your bones grow in harmony with your muscles, the reason your body can adjust to the slow changes of season and age. Even now, as you read, your endocrine system is at work. Somewhere in your body, a gland is releasing a molecule into your blood. That molecule is traveling quietly, carried by the pulse of your heartbeat, seeking the cells prepared to hear its message. When it finds them, it will shape their behavior in ways that support the whole, without fanfare, without urgency, but with steady, lasting purpose. 8. Neurotransmitters and nerve cell conversations. If hormonal communication is like sending a carefully prepared letter across the country, then nerve cell communication is like leaning close to speak in someone's ear. It is immediate, precise, and perfectly suited for messages that cannot wait. In the body, this is the work of neurotransmitters, the chemical messengers of the nervous system. Nerve cells, or neurons, are the body's communication specialists. Each one is shaped to send and receive information with remarkable speed. A neuron can be thought of as a living electrical cable, carrying signals from one end to the other in fractions of a second. Yet for all their speed, neurons do not actually touch each other. Between the end of one neuron and the beginning of the next is a tiny gap called a synapse, so small that it is measured in thousandths of a millimeter, yet so important that all nerve communication depends on it. When a neuron has a message to send, it begins with an electrical impulse, known as an action potential. This pulse travels down the length of the neuron, carried by the movement of charged particles called ions, across the cell's membrane. The journey may be long, from the spinal cord to the tip of your toe, or from the base of the brain to the muscles that move your eyes. But the impulse moves at great speed, arriving at the end of the neuron in less than the blink of an eye. At this point, the nature of the message changes. The electrical signal cannot jump the synapse on its own. Instead, it triggers the release of neurotransmitters, small molecules stored in tiny packages called vesicles. These molecules spill into the synaptic gap and drift the short distance to the next cell. On the surface of that receiving cell are receptors, proteins shaped perfectly to recognize and bind to specific neurotransmitters. When the binding occurs, it starts a new chain of events inside the receiving cell, which may itself send an electrical impulse forward or begin some other action entirely. This conversion from electrical to chemical and back again allows the nervous system to combine speed with control. The synapse is not just a bridge, but a place where messages can be adjusted, strengthened, weakened, or even blocked entirely. This flexibility is what gives the brain and nervous system their ability to learn, adapt, and make fine distinctions in response to the world. Different neurotransmitters carry different kinds of messages. Some, like glutamate, encourage the receiving cell to fire its own impulse, spreading the message onward. Others, like gamma-aminobutyric acid, GABA, have the opposite effect, quieting activity and preventing overstimulation. Still, others serve more specialized roles. Dopamine can influence motivation and reward. Serotonin helps regulate mood and sleep, and acetylcholine controls the contraction of muscles. Each neurotransmitter has its own tone and character, its own particular influence on the cells that receive it. The precision of neurotransmitter signaling is remarkable. Because neurotransmitters are released only at synapses and broken down or taken back up almost immediately, their effects are localized and brief. This allows the nervous system to respond in real time to changes in the environment. A faint sound in the distance can prompt the rapid release of neurotransmitters that activate muscles and sharpen attention. A sudden loss of balance can set off a chain of signals that restore your footing before you even think about it. This speed and specificity make neurotransmitter communication the primary means by which the brain coordinates fast, complex actions. Every thought, every movement, every sensation you feel is shaped by countless neurotransmitter exchanges happening in parallel across billions of synapses. The patterns of these exchanges are as individual as fingerprints, shifting moment by moment with experience, mood, and activity. Although neurotransmitter signaling is fast, it is not purely mechanical. The nervous system can strengthen or weaken connections between neurons depending on how often they are used, a process known as synaptic plasticity. When two neurons communicate frequently, the efficiency of their connection can increase, making it easier for signals to pass. This is part of the biological basis for learning and memory. Just as repeated conversation strengthens understanding between people, repeated communication strengthens the pathways between nerve cells. Neurotransmitter signaling also interacts with other forms of communication in the body. Nerve impulses can trigger the release of hormones, linking the nervous and endocrine systems. Hormones, in turn, can influence the sensitivity of neurons to neurotransmitters, adjusting how readily they respond. In this way, the body blends the rapid moment-to-moment control of neurotransmission with the slower, longer-term guidance of hormonal signaling. The importance of neurotransmitters extends beyond voluntary actions. They are equally vital for the automatic processes that keep you alive: the steady beating of the heart, the expansion and contraction of the lungs, the regulation of digestion. In the brainstem and spinal cord, networks of neurons use neurotransmitters to maintain these rhythms without requiring conscious thought. You breathe, your heart pumps, and your body adjusts to changing needs, all through the quiet work of nerve cell conversations. If we could witness these exchanges at their true scale, we might imagine a city lit by countless points of light. Each neuron would glow faintly along its length, then flare at the synapse, as an electrical pulse arrived. In that moment, tiny packets of neurotransmitters would spill into the space between, like sparks leaping from one filament to another. The receiving cell would light up in turn, passing the signal onward, creating waves of illumination that ripple through the network. In such a vision, the nervous system becomes a living constellation, each connection a star linked by invisible pathways. And like the stars, these connections form patterns, constellations of thought, sensation, and action that shift and reform as you experience the world. Every time you speak, listen, move, or remember, the constellations change, redrawn in the subtle flicker of neurotransmitter exchanges. Even in sleep, this activity does not cease. The brain continues to send and receive messages, processing memories, maintaining vital functions, and preparing for the day to come. The conversation of nerve cells is unending, a hum beneath every waking moment and every dream. Neurotransmitter signaling is the nervous system's gift to the body, a way of responding instantly to the world while retaining the flexibility to adapt and learn. It is the reason you can catch a falling object, respond to a sudden sound, or recall a familiar face in an instant. It is the voice of immediacy, spoken in a chemical language that travels only a few millionths of a meter, yet binds the body and mind into one responsive whole. In the delicate interplay between neurotransmitters and their receptors lies the foundation of awareness, thought, and movement. And though the messages themselves are fleeting, the patterns they create form the ongoing conversation that is, in many ways, the experience of being alive. 9. Electrical signals in the body. Within the quiet, unseen world of your body, there exists a current of information that moves not with molecules floating in fluid, but with the swift, invisible motion of charged particles. These are the body's electrical signals, pulses of energy that carry messages at remarkable speed, guiding everything from the beat of your heart to the movement of your eyes. Electrical signals are possible because of the unique properties of cell membranes. Every living cell maintains a delicate separation between its inner environment and the space outside, and across this thin barrier, there exists a subtle difference in electrical charge. This difference, called the membrane potential, is created by the uneven distribution of charged ions, mainly sodium, potassium, calcium, and chloride on either side of the membrane. Cells maintain this balance through the careful work of ion pumps and channels, proteins embedded in the membrane that allow certain ions to pass in or out. In many cells, the membrane potential is steady, serving simply as a background condition, but in specialized cells, such as neurons and muscle fibers, that potential can change rapidly. When a stimulus reaches a certain threshold, ion channels open in sequence, allowing charged particles to rush across the membrane. This sudden movement changes the electrical balance, creating a brief pulse of energy called an action potential. An action potential is not a random burst. It is a signal, a clear and measurable event that travels along the length of the cell. In neurons, it carries information from the cell body down to the synaptic terminals, where the signal will be passed on chemically to the next cell. In muscle fibers, the action potential triggers the release of calcium inside the cell, initiating contraction. The beauty of electrical signaling lies in its speed. Because the signal is a wave of ionic change, rather than a molecule moving through space, it can travel far more quickly than chemical messengers alone. In some nerve fibers, especially those coated in insulating myelin, action potentials can leap from one exposed section to another, a process called saltatory conduction. This allows impulses to travel at speeds up to hundreds of kilometers per hour, ensuring that the body can respond instantly to the world. Electrical signals are not limited to neurons. In the heart, specialized pacemaker cells generate rhythmic action potentials that spread through the heart muscle via gap junctions. This electrical wave coordinates the contraction of the heart chambers, producing the steady, unified beat that pumps blood throughout the body. The same principle operates in certain smooth muscles, such as those of the digestive tract, where waves of electrical activity coordinate the slow, steady contractions that move food along. Even plants make use of electrical signals. In response to touch or injury, some plants generate action potentials that travel through their tissues, triggering changes such as the rapid closing of a venous flytrap or the folding of sensitive leaves. While these signals move more slowly than in animals, they show that the language of electrical change is not limited to the animal kingdom. The generation of an electrical signal is only the beginning of the story. For the message to have meaning, it must be received and acted upon by other cells. This often involves a translation from electrical form to chemical form, as in the synapses of the nervous system. In other cases, the electrical change directly triggers a physical action, such as muscle contraction. In both situations, the pattern, timing, and frequency of the signals carry important information. A train of rapid pulses may produce one effect, while slower, intermittent pulses produce another. The precision of electrical signaling is remarkable. The same basic mechanism, the flow of ions across a membrane, can be shaped into countless patterns, each with its own meaning. In the auditory system, for example, the pattern of action potentials in nerve fibers encodes the pitch and volume of a sound. In the visual system, electrical signals carry information about light intensity, color, and movement from the retina to the brain. One of the most fascinating aspects of electrical communication is its universality. Although the details vary, the basic principles are the same in humans, other animals, and even certain plants and single-celled organisms. The same flows of sodium and potassium, the same opening and closing of channels, the same wave-like propagation of charge, these are found across the tree of life, evidence of a shared evolutionary heritage stretching back billions of years. Electrical signals are also deeply connected to the rhythms of life. In the brain, large networks of neurons produce oscillations repeating patterns of electrical activity that correspond to different states of consciousness. Slow, high-amplitude waves dominate during deep sleep, while faster rhythms emerge during wakefulness and focused attention. These patterns are not just byproducts of brain activity. They help coordinate the timing of signals across distant regions, much like a conductor guiding the sections of an orchestra. In the heart, the rhythm is steadier but no less essential. The electrical pulses generated by the pacemaker cells are influenced by signals from the nervous system and hormones in the blood, allowing the heartbeat to adjust to activity, rest, or emotion. In this way, electrical signals link the heart's mechanical work to the broader needs of the body. If we could see electrical signals in real time, the body might appear as a glowing network. Pulses of light would flash along the lengths of neurons, ripple across the heart muscle, and travel in coordinated waves through smooth muscle layers. The brightness and speed of these pulses would change constantly, responding to the shifting demands of movement, thought, and sensation. Even now, as you sit quietly, electrical signals are at work. They carry the impulse from your brain to your diaphragm with each breath, adjust your posture through subtle muscle contractions, and transmit the sensory impressions from your eyes and ears to your mind. They are the invisible threads that connect every part of your body in the present moment, ensuring that you remain not just alive, but responsive and aware. Electrical signals represent the body's swiftest means of communication. They complement the slower, more enduring effects of chemical messengers, allowing for a balance between immediate reaction and long-term regulation. Together, they form a dual language, one spoken in the instant flashes of charged particles, the other in the slower currents of molecules drifting through fluids. In the end, electrical signaling is more than a biological process. It is the pulse of life itself, a ceaseless current that runs through the living body from birth to death. It is the reason the heart keeps beating, the reason the muscles respond to thought, the reason the senses can connect you to the world in the very moment you encounter it. It is a silent, flowing language, as old as the first spark of life, and as constant as the rhythm of your own breath. 10. How cells listen to signals. Every message in the body, whether it travels as a drifting molecule, a racing electrical impulse, or a direct physical touch between neighboring cells, is only meaningful if it is heard. A signal without a listener is like a letter never opened, a whisper in an empty room. The true power of communication in living systems lies not only in sending the right message, but also in the ability of the recipient to recognize and interpret it. In the world of cells, listening is the work of structures called receptors. A receptor is a specialized protein crafted to detect a particular type of signal. Some are embedded in the outer membrane of the cell, facing outward into the surrounding environment. Others lie deep inside the cell, waiting for messengers that can pass through the membrane to reach them. Each receptor is built to recognize only certain molecules or physical cues, much like a lock designed for a specific key. This selectivity ensures that a cell responds only to the messages intended for it, ignoring the countless other signals that may pass by. When the correct signal, the right key, binds to its receptor, the receptor changes shape. This subtle shift is not just a physical movement, it is the beginning of a translation process. The binding event triggers a series of internal changes, converting the presence of the external signal into meaningful instructions inside the cell. These instructions may tell the cell to begin producing a certain protein, to open an ion channel, to divide, or to prepare for movement. Different types of receptors are tuned to different kinds of messages. Ion channel receptors, for instance, control the passage of charged particles across the membrane. When they bind to their signal, they open briefly, allowing ions to flow in or out, changing the electrical state of the cell. This is the basis for many rapid responses in nerve and muscle cells. G-protein-coupled receptors detect a wide range of molecules, from hormones to sensory cues like light and smell, and pass the message inward through complex signaling cascades. Enzyme-linked receptors trigger chemical reactions inside the cell when activated, often leading to changes in metabolism or growth. Intracellular receptors, found inside the cytoplasm or nucleus, respond to signals that can pass directly through the membrane, such as steroid hormones. Listening is not passive. Cells can adjust how closely they listen to a particular signal by changing the number of receptors they have or altering their sensitivity. If a signal is rare but important, a cell may increase the number of receptors to catch every possible molecule. If a signal is constant and overwhelming, the cell may reduce its receptors to avoid overstimulation. This ability to fine-tune sensitivity is essential for maintaining balance. Without it, a cell could become unresponsive to critical cues or react too strongly to minor fluctuations. The arrangement of receptors across the cell surface can also shape how a message is heard. In some cases, receptors cluster together when activated, reinforcing the strength of the incoming signal. In others, they are spread widely, allowing the cell to sample a larger area. The spatial organization of receptors can create gradients of activity inside the cell, guiding movement toward or away from a source of signal. Receptors do not work in isolation. Often, multiple receptors will detect different parts of a complex message, with the cell integrating these inputs to decide on the most appropriate response. For example, an immune cell might need to detect both the presence of a foreign molecule and a signal from another immune cell before launching a defensive action. This layered decision-making reduces the chance of false alarms and ensures that responses are proportional to the situation. The process of receiving a signal and turning it into action is known as signal transduction. Once the receptor is activated, it sets off a chain of molecular events inside the cell. These events may involve second messengers, small molecules that spread the signal internally, as well as changes in the activity of enzymes and the rearrangement of cellular structures. The signal can be amplified at each step, so that a single binding event at the surface may lead to a large and coordinated response throughout the cell. Listening is also about timing. Some receptors are tuned to respond quickly, triggering actions within milliseconds. Others work more slowly, guiding changes that unfold over minutes, hours, or even days. The duration of receptor activation influences how long the cell's response lasts. A brief activation may produce a temporary effect, while prolonged activation can lead to lasting changes in the cell's behavior or identity. In addition to detecting chemical messengers, some receptors are designed to sense mechanical forces, temperature changes, or light. In your skin, mechanoreceptors detect the stretch and pressure of touch, while thermoreceptors respond to warmth or coolness. In your eyes, light-sensitive receptors in the retina capture photons and initiate the complex process of vision. All of these are examples of the cell's ability to listen not just to internal chemical signals, but also to the world beyond the body's boundaries. This act of listening is deeply ancient. Even the simplest single-celled organisms have receptors, allowing them to detect food sources, avoid harmful substances, and interact with others of their kind. Over billions of years, these sensing abilities have become more elaborate, supporting the development of multicellular life and the intricate coordination of tissues and organs. If we could see a receptor at work, it might appear as a small, complex structure resting on the cell's surface, swaying gently in the fluid environment. A signal molecule approaches, shaped perfectly to fit. Upon contact, the receptor shifts, and deep within the cell, molecular machinery begins to move. The signal is passed inward, transformed from an external presence into an internal instruction, ready to guide the cell's next step. Even as you sit quietly, your cells are listening. They hear the soft call of hormones in your blood, the electrical murmurs of nearby neurons, the local chemical signals of their neighbors. They hear the stretch of muscle, the temperature of your skin, the presence of nutrients. Each of these messages is interpreted, weighed, and answered in ways that keep you balanced, responsive, and alive. In this way, listening is as vital as speaking. Without receptors to detect and understand signals, the body's messages would drift without purpose, like unclaimed letters in an endless sea. But with receptors in place, every message has a chance to be heard, understood, and acted upon, a conversation that never ends, a dialogue that binds all the cells of your body into one living whole. 11. Signal transduction, turning messages into action. When a signal reaches a cell and binds to its receptor, it is like the moment a letter is opened. The message is in hand, but it still must be read, understood, and acted upon. This process of translating the arrival of a signal into a meaningful change within the cell is called signal transduction. It is the bridge between hearing a message and responding to it, the sequence of events that turns a single binding event on the cell's surface into a cascade of activity deep inside. Signal transduction is not a single step. It is a journey, a relay race in which the baton is passed from one molecule to another, each stage moving the signal inward and often amplifying it along the way. At the starting point is the receptor, freshly activated by its specific signal. This activation changes the receptor's shape or chemical state, creating a trigger for the next player in the chain. Sometimes this next player is a protein directly attached to the receptor. Other times, the receptor activates a nearby molecule, setting the chain reaction in motion. One of the most remarkable features of signal transduction is amplification. A single activated receptor can influence many molecules, and each of those can, in turn, influence many more. This means that a very small signal, Perhaps just a few molecules binding to receptors can produce a large and coordinated response throughout the cell. This amplification is essential for sensitivity. It allows cells to detect and respond to signals even when those signals are present at extremely low levels. The steps in a signal transduction pathway can be highly varied, but many involve changes in protein shape, activity, or location. Some proteins act as switches, flipping from an off state to an on state when they receive the right cue. Others act as messengers, moving from one part of the cell to another to deliver the signal. Still others serve as builders, creating small molecules known as second messengers that carry the signal deeper into the cell. These second messengers, such as cyclic AMP, calcium ions, or inositol phosphate, spread rapidly, influencing a wide range of targets at once. The speed of a signal transduction pathway depends on its purpose. Some are designed for speed, activating in milliseconds to guide urgent actions like muscle contraction or sensory perception. Others move more slowly, guiding processes like cell division, gene expression, or metabolic adjustments that unfold over hours or days. The timing is not arbitrary, it matches the demands of the task at hand. One of the key strengths of signal transduction is its ability to integrate multiple signals at once. Inside the cell, different pathways can meet, intersect, and influence each other. This allows the cell to consider several pieces of information before deciding how to act. For example, a liver cell might receive a signal from insulin encouraging it to store glucose, while at the same time receiving a signal from adrenaline, prompting it to release glucose for energy. The outcome will depend on the balance between these signals and the cell's current state. This kind of integration ensures that the cell's response is not rigid but flexible, adapting to the overall needs of the organism. Signal transduction is also precise in space. In some cases, the molecular changes it triggers happen only in a specific part of the cell near the membrane, around the nucleus, or along certain structural fibers. This localization allows the cell to focus its response exactly where it is needed, avoiding unnecessary disruption to other processes. Another important feature is reversibility. A cell's environment is constantly changing, and once a signal has done its work, the pathway that carried it must be switched off. Enzymes may break down second messengers, deactivate proteins, or remove chemical tags that had altered protein function. Without these reset mechanisms, signals could linger too long, leading to confusion or harm. The ability to turn a pathway both on and off is essential for maintaining balance. In living systems, the consequences of signal transduction are as varied as life itself. It may lead to the opening of an ion channel, altering the cell's electrical state. It may switch on genes in the nucleus, leading to the production of new proteins. It may reorganize the cell's internal scaffolding, changing its shape or movement. It may activate enzymes that speed up or slow down chemical reactions. In many cases, it does several of these things at once, coordinating a complex and purposeful response. If we could watch signal transduction inside a single cell, it might appear as a series of ripples spreading inward from the membrane. At the point where the signal arrived, proteins would shift their shapes and latch onto new partners. Tiny messenger molecules would dart through the cytoplasm, touching off reactions in multiple places at once. Structures would rearrange, enzymes would awaken, and the nucleus might begin quietly copying instructions into RNA. All of this would happen without a sound, in a space smaller than the width of a human hair, yet with an order and elegance that rival any human-made network. This process is not just a marvel of biology, it is also a safeguard. Because the signal is passed through multiple steps, each one can be checked, modified, or stopped if needed. This ensures that false alarms do not set off costly or dangerous responses. In some cases, the pathway includes built-in brakes proteins that will shut the system down if the signal is too weak or prolonged. Signal transduction is ancient, found in all living organisms from bacteria to humans. In the earliest cells, Primitive receptors and signaling molecules allowed organisms to detect light, nutrients, and toxins. Over billions of years, these systems have grown in complexity, allowing the cells of multicellular organisms to coordinate their activities with extraordinary precision. Despite their diversity, the core principle remains the same. A signal outside the cell is transformed into a response inside the cell, linking perception to action. Even now, as you breathe and your heart beats steadily, countless signal transduction events are happening within you. Hormones binding to their receptors are setting off cascades in the liver, adjusting metabolism. Neurotransmitters are triggering rapid changes in neurons, guiding your thoughts and movements. Growth factors are quietly influencing the repair of tissues, and immune signals are shaping the readiness of your defenses. Each of these processes begins with a signal received, then translated into the language of action by the intricate machinery of the cell. Signal transduction is the art of turning awareness into behavior, of transforming recognition into response. It is the means by which a cell moves from knowing to doing, from potential to reality. Without it, the messages that flow through the body would be silent, seen, but never understood, felt, but never answered. With it, life becomes a constant, meaningful conversation, carried out in countless tiny steps, each one leading the cell closer to the action it needs to take. 12. Second messengers inside the cell. When a signal arrives at a cell and binds to its receptor, the first step in communication is complete. The message has been received. But in many cases, the real work of spreading that message through the cell is only beginning. The receptor, activated by its external signal, now passes the word inward, often through the help of small, highly mobile molecules known as second messengers. These second messengers are the couriers of the cell's internal communication system. They do not exist to interpret the message themselves. Rather, they carry it rapidly and efficiently to multiple parts of the cell, ensuring that the response is coordinated. In this way, they amplify the original signal, allowing a single binding event at the surface to influence a vast network of internal processes. Second messengers are small and diffuse quickly, moving through the fluid interior of the cell with ease. Because they can reach many targets at once, they are especially powerful in creating widespread effects. Among the most common and important are cyclic AMP, CAMP, calcium ions, inositol phosphates, and cyclic GMP, CGMP. Each plays a unique role, and each can set off its own distinct cascade of events. One of the most widely studied second messengers is cyclic AMP, often produced when a signal activates a G protein-coupled receptor at the membrane. CAMP is created from ATP, the cell's main energy molecule, by an enzyme called adenylate cyclase. Once formed, CAMP spreads through the cytoplasm, activating a variety of proteins, most notably protein kinase A. This enzyme can then modify other proteins by adding phosphate groups to them, altering their activity in ways that help bring about the cell's response. Calcium ions are another key second messenger, and they are remarkable for the speed and versatility of their effects. Normally, calcium levels in the cytoplasm are kept very low compared to the outside of the cell or within certain internal compartments. When a signal calls for action, channels open to allow calcium to flood into the cytoplasm. This sudden rise in calcium concentration can trigger a wide range of responses: muscle contraction, secretion of hormones or neurotransmitters, changes in metabolism, or even the activation of certain genes. Because calcium signals can be brief or sustained, local or global, they can carry a great deal of nuanced information. Inositol phosphates, such as inositol trisphosphate, IP3, work closely with calcium in many signaling pathways. IP3 is produced when a membrane lipid is cleaved by an activated enzyme in response to a signal. It then diffuses through the cytoplasm to bind receptors on the endoplasmic reticulum, an internal membrane system that stores calcium. This binding causes the release of calcium into the cytoplasm, adding another layer of amplification to the signal. Cyclic GMP, or cGMP, serves as a second messenger in processes ranging from the relaxation of smooth muscle to the detection of light in the eye. In the retina, for example, cGMP levels control the opening and closing of ion channels in photoreceptor cells, allowing the conversion of light into electrical signals for vision. In blood vessels, cGMP mediates the effects of nitric oxide, leading to the relaxation of smooth muscle and increased blood flow. What makes second messengers so effective is not just their mobility, but their ability to multiply the impact of the original signal. A single activated receptor might produce dozens or hundreds of second messenger molecules. Each of those, in turn, can activate multiple target proteins, creating a branching network of effects that reach deep into the cell. This amplification means that even a faint external signal can produce a strong and coordinated response. Second messengers also contribute to the flexibility of cellular communication. Because different pathways can share the same second messenger, the cell can integrate multiple signals that arrive at once. For instance, calcium may be released in response to one hormone, but also in response to a neurotransmitter. The combined calcium signal reflects the influence of both inputs, allowing the cell to weigh their importance and respond accordingly. Timing is another key aspect of second messenger action. These molecules are not meant to linger indefinitely. Once they have done their job, they are broken down or removed to reset the system. CAMP, for example, is degraded by enzymes called phosphodiesterases. Calcium is pumped back into storage compartments or out of the cell. This clearance ensures that the signal is temporary, giving the cell the freedom to respond to new messages without interference from old ones. The patterns of second messenger signals can be just as meaningful as their intensity. A brief pulse of calcium may trigger one set of responses, while a longer, sustained increase may trigger another. Repeated bursts of CAMP may have different effects than a single prolonged rise. Cells can read these temporal patterns and tailor their actions accordingly, much like how the rhythm of spoken words can change their meaning. Second messengers also operate in distinct spaces within the cell. While they can diffuse freely, their production and effects are often localized to specific regions. This creates microdomains of signaling activity, allowing for precise control. In a neuron, for example, calcium signals in the synaptic terminal can trigger neurotransmitter release without affecting calcium-sensitive processes elsewhere in the cell. If we could see second messengers at work, it might appear as a sudden bloom of color inside the cell, spreading outward from the receptor, like ink in water. Tiny ripples of calcium would appear and fade. Waves of CAMP would rise and disperse. Pockets of inositol phosphate would spark the release of stored calcium. These events would occur in patterns, bursts, waves, gradients, each carrying a specific meaning for the cell's machinery. In the living body, second messengers are at work constantly. When you step into a bright room, second messengers in your eye's photoreceptor cells adjust your sensitivity to light. When you stand up quickly, second messengers in your blood vessels help them constrict to maintain your blood pressure. When you learn something new, second messengers in your brain help strengthen the connections between neurons, laying the groundwork for memory. The role of second messengers is both humble and profound. They do not decide what the signal means or what the final action should be. Instead, they ensure that the message, once received, is delivered swiftly and widely to the right places within the cell. They are the runners in the relay, the couriers that turn a single moment of recognition at the cell's surface into a sweeping, coordinated change in the cell's life. Without them, many of the body's most important signals would never reach their full potential. With them, a single touch, thought, or chemical cue can ripple through the intricate machinery of life, shaping behavior, guiding growth, and maintaining the delicate balance that makes living systems possible. 13. amplifying small signals. In the intricate world of cellular communication, not every message arrives as a strong, undeniable signal. Often, the earliest hint of change begins as something faint, a single molecule drifting through the fluid between cells, a tiny fluctuation in electrical potential, a subtle change in the concentration of ions. Left alone, such a small event might pass unnoticed, too weak to cause meaningful action. Yet within living systems, even these delicate beginnings can be transformed into decisive, coordinated responses. This transformation is the art of signal amplification. Signal amplification allows a cell to take the smallest spark of information and expand it into a wave of activity that can influence large areas of the cell or even entire tissues. Without amplification, a weak signal might be lost in the constant background of cellular activity. With amplification, the same weak signal can set off a cascade of changes, ensuring that important messages are never missed. The process begins at the moment a receptor detects its specific signal. Whether that receptor sits on the cell's surface or deep within its interior, its activation is the first step in a chain of events. This chain, or pathway, is carefully arranged so that each step activates not just one new molecule, but many. In this way, a single activation is multiplied at every stage. Consider the binding of a hormone to its receptor on the cell surface. One activated receptor can, in turn, activate multiple molecules of a G protein, a kind of molecular switch. Each of these activated G proteins can then stimulate an enzyme such as adenylate cyclase, which can produce hundreds of molecules of the second messenger cyclic AMP. Each molecule of CAMP can activate a protein kinase, which can then modify dozens of target proteins within the cell. In only a few steps, the original signal has been magnified thousands or even millions of times. Amplification is not limited to chemical pathways. In electrical signaling, a small change in membrane potential can open ion channels, allowing a rush of charged particles into the cell. This movement of ions can then open additional channels in nearby regions, spreading the electrical change rapidly. In neurons, this is the basis of the action potential, which can travel long distances while maintaining its strength. One of the key advantages of amplification is efficiency. The body does not need to flood the system with signal molecules in order to produce a strong effect. Instead, it relies on the design of the signaling pathways to make the most of what is available. This is especially important for hormones and other chemical messengers that must be produced in limited amounts and transported over long distances. Amplification also contributes to sensitivity. Cells can detect and respond to signals present in incredibly low concentrations, sometimes only a few molecules per cell, because the pathways that process those signals are built to multiply their effects. This sensitivity is vital for processes such as detecting faint light in the retina, sensing small changes in blood chemistry, or responding to early signs of infection. In the visual system, amplification is taken to extraordinary levels. In the rod cells of the eye, a single photon of light can activate a receptor called rhodopsin. This, in turn, activates hundreds of molecules of a G protein called transducin, which then activate hundreds more molecules of an enzyme that breaks down cGMP. The resulting changes close ion channels in the cell membrane, altering the electrical state of the cell, and ultimately producing a signal that the brain interprets as light. In this way, the eye can detect the faintest glimmer in near darkness. Amplification, however, is a carefully controlled process. Left unchecked, it could lead to excessive responses that disrupt the delicate balance of the body. To prevent this, cells include regulatory mechanisms at multiple points in the pathway. Enzymes can inactivate key signaling proteins, degrade second messengers, or close ion channels, ensuring that the amplification stops once the signal has been processed. Feedback loops can adjust the strength of amplification in response to changing conditions, keeping the system stable. There is also a kind of elegance in how amplification is distributed within the cell. In some cases, the signal spreads broadly, activating processes throughout the cell. In others, it is confined to specific regions, producing strong local effects without disturbing other areas. This spatial control allows cells to direct their responses precisely where they are needed at the leading edge of a migrating cell, for example, or in the synaptic terminal of a neuron. Amplification is not always about creating a strong singular response. Sometimes, it is about ensuring reliability. In a noisy environment filled with competing signals, the cell needs a way to make sure that genuine messages rise above the background. By amplifying those messages early in the pathway, the cell increases the likelihood that they will be recognized and acted upon. If we could see amplification happening in real time, it might look like the spreading ripples from a single drop of water falling into a still pond. At first, there is only the point of contact, a small disturbance at the surface. Then, circles spread outward, touching larger and larger areas, each wave carrying the energy of that first small event. Inside the cell, the ripples are made of molecules, proteins, and ions, each step extending the influence of the original signal. In the context of life, amplification is part of what allows small causes to have meaningful effects. It is the reason a faint scent can trigger a cascade of memories, the reason a soft touch can lead to a complex emotional response, the reason the body can react quickly to subtle changes in its environment. It ensures that even the gentlest message can be heard clearly, and that important information is never lost simply because it arrives quietly. Amplification also plays a role in learning and adaptation. In the nervous system, repeated activation of certain pathways can make amplification more efficient over time, strengthening the connections between neurons. This is one of the ways experiences are encoded into memory, as repeated signals become easier to trigger and more effective in producing a response. In the immune system, amplification allows a small number of antigen-presenting cells to set off a large-scale defensive response. A few activated cells can release signals that recruit and activate many others, rapidly building a coordinated defense against a pathogen. Without this ability, the immune system would be far slower and less effective. Even in sleep, amplification is at work. As your body repairs itself, small chemical cues trigger cascades that promote tissue growth, adjust metabolism, and strengthen neural connections. These processes do not require dramatic signals, only the quiet but powerful multiplication of small ones. Amplifying small signals is one of life's most resourceful strategies. It turns scarcity into sufficiency, weakness into strength, and faint beginnings into decisive actions. It is a reminder that in living systems, impact is not determined only by the size of the initial push, but by the ability to carry that push forward, to multiply it, and to let it grow into something far greater than itself. 14. Fine-tuning the response. In the intricate language of cells, receiving and amplifying a signal is only part of the story. Once a message has been heard and set into motion, the next challenge is to respond in a way that is appropriate, balanced, and precise. Too weak a response may fail to address the need. Too strong a response may cause harm. This delicate balance is achieved through the fine-tuning of cellular responses, the careful adjustment of strength, timing, and location of activity to match the situation at hand. Fine-tuning begins with feedback. In many signaling pathways, the molecules activated by the signal can, in turn, influence the very pathway that created them. This feedback can be negative, dampening the response once it has reached the necessary level, or positive, briefly boosting it when more action is required. Negative feedback is particularly important for preventing overreaction. Once a sufficient amount of work has been done, enough enzymes activated, enough ions moved, enough genes turned on the system, applies its own brakes, allowing the response to ease back toward a resting state. Positive feedback, on the other hand, is used when a rapid and decisive action is needed. For example, in blood clotting, once a small clot begins to form, certain clotting factors accelerate their own activation, leading to a burst of activity that seals the wound quickly. But even in such cases, other mechanisms stand ready to slow or stop the process once the danger has passed, preventing excessive clotting that could block healthy blood flow. Another aspect of fine-tuning lies in controlling the sensitivity of the cell to the signal itself. Cells can adjust how many receptors they display on their surfaces, effectively changing the volume of the signal they hear. If a signal is weak but important, a cell may increase its number of receptors, heightening its awareness. If the signal is constant or overwhelming, the cell may reduce its receptors to avoid overstimulation, a process known as receptor down regulation. This adjustment can occur over minutes, hours, or days, depending on the cell's needs. Fine tuning also involves regulating the duration of the response. Some signals are best answered with a quick burst of activity, while others require a sustained effort. Cells achieve this control through the timing of second messenger production, the lifespan of activated proteins, and the speed with which enzymes break down signaling molecules. A short, sharp calcium pulse might trigger the release of neurotransmitters, while a longer, steadier calcium signal could influence gene expression and growth. Spatial precision is another layer of regulation. The effects of a signal can be confined to specific areas within the cell, ensuring that the response happens exactly where it is needed. For example, in a nerve cell, calcium signals at the synaptic terminal can trigger neurotransmitter release without affecting calcium-sensitive processes in the cell body. This compartmentalization allows for multiple signaling events to occur in the same cell at the same time without interfering with each other. The interplay between different signaling pathways is also a powerful tool for fine-tuning. A single cell often receives multiple signals at once, and the pathways they activate can interact in complex ways. Some signals may reinforce each other, leading to a stronger combined effect. Others may counterbalance, ensuring that one pathway's activation prevents another from going too far. This crosstalk between pathways allows the cell to integrate information from its environment and choose the most balanced course of action. In the immune system, fine-tuning is vital for distinguishing between helpful and harmful responses. Immune cells must react strongly enough to eliminate threats, but not so strongly that they damage healthy tissue. Here, regulatory molecules act as moderators, tempering the activity of immune cells when the threat subsides. A failure of this moderation can lead to autoimmune disorders, where the immune system attacks the body's own cells. In the nervous system, fine-tuning allows for the subtle control of thought, sensation, and movement. Neurons can adjust the strength of their connections with one another, a property known as synaptic plasticity, to match the importance of a signal. Frequently used pathways can be strengthened, making them more responsive, while less used pathways can be allowed to fade. This adaptability underlies learning, memory, and the brain's ability to refine its performance over time. Even in processes as automatic as heartbeat regulation, fine tuning is essential. The heart's pacemaker cells set the basic rhythm, but signals from the nervous system and hormones can speed it up or slow it down. These adjustments are finely calibrated so that the heart delivers the right amount of blood for the body's needs at any given moment, whether in the quiet of rest or the urgency of physical exertion. If we could observe fine-tuning at work inside a cell, we might see waves of activity rising and falling like the tides, their shapes and heights adjusted by countless small decisions. Enzymes would be switching on and off, receptors appearing and disappearing from the membrane, messenger molecules being produced and then cleared away. Each adjustment would be subtle in itself, but together they would create a balanced and harmonious response, shaped precisely to fit the circumstances. This process is not rigid or mechanical. It is dynamic, responsive, and continuous. Fine tuning happens not only at the end of a signaling event, but throughout its course. The cell is always measuring its own activity, sensing the flow of messages, and making small corrections to keep everything aligned. In this way, the response is never too much, never too little, but just enough to serve its purpose. The importance of fine tuning extends to the organism as a whole. In your body right now, countless adjustments are being made in parallel hormone levels, shifting slightly to balance energy use, blood vessels narrowing or widening to guide circulation, neurons adjusting their sensitivity to keep sensory information clear and steady. These are not dramatic events, but their quiet constancy is what keeps life running smoothly. Fine-tuning is the art of precision in biology. It transforms the blunt force of amplification into a refined, measured action. It ensures that signals lead not to chaos but to coordination, not to waste but to efficiency. It allows the body to operate with the grace of a well-practiced orchestra, each instrument playing its part at the right volume, the right tempo, and in perfect harmony with the others. Without fine tuning, the systems of life would be prone to overreaction and instability. With it, even the most powerful signals can be shaped into calm, purposeful action. It is the quiet discipline of living systems, the constant, careful shaping of responses, so that every action serves its purpose, and no more. 15. Coordinated activity in tissues and organs. A single cell is remarkable on its own. It can sense its surroundings, process information, and carry out complex tasks with precision. Yet, in the living body, no cell exists in true isolation. It is part of a vast community, and its life gains greater meaning when seen as part of a larger whole, the tissue or organ to which it belongs. The quiet conversation between cells is what allows this larger whole to function not as a jumble of parts but as a unified living system. Within any given tissue, thousands or even millions of cells work together toward a shared purpose. In muscle tissue, that purpose may be movement. In the liver, it may be the careful management of nutrients and detoxification. In the skin, it is protection and renewal. Whatever the task, coordination is essential. A single cell acting out of step could weaken the performance of the entire tissue. For this reason, cells constantly exchange signals, aligning their activity with that of their neighbors. In muscle tissue, coordination is especially visible. When you decide to move your arm, nerve impulses travel to muscle fibers, prompting them to contract. But the strength and smoothness of that motion depend on more than just one muscle fiber responding. The fibers must contract together, in the right sequence, with the right amount of force. This is achieved through both electrical signals that spread quickly between fibers and chemical messengers that adjust their readiness to act. Even in the heart, where contractions happen without conscious control, the cells that make up the heart muscle cardiomyocytes beat in perfect unison, thanks to direct electrical connections through gap junctions. The same principle applies in glands and organs responsible for secretion. In the pancreas, for example, clusters of cells known as islets of Langerhans release hormones such as insulin and glucagon into the bloodstream. These hormones regulate blood sugar levels across the body. Yet the secretion of these hormones is not random. It is carefully timed and balanced, with each cell in the cluster adjusting its output in response to signals from its neighbors. This local coordination ensures that the hormones are released in just the right amounts to maintain stability. In tissues that act as barriers, such as the lining of the gut or the surface of the skin, coordination ensures that the protective layer remains intact. Cells at the surface are constantly being shed and replaced. Signals from deeper layers guide this renewal, telling new cells when to divide, when to move upward, and when to mature into their final protective form. If this process were not synchronized, Gaps could appear in the barrier, leaving the body vulnerable. Coordination also extends to the sharing of resources. In many tissues, cells pass nutrients, ions, and even small signaling molecules directly from one to another through junctions in their membranes. This sharing allows cells in less favorable positions, perhaps farther from a blood vessel, to receive what they need to survive and perform their work. In this way, the tissue functions as a collective, distributing resources where they are most needed. In the nervous system, coordination occurs on an even grander scale. Networks of neurons form circuits that handle everything from sensing light and sound to controlling movement and regulating mood. The effectiveness of these circuits depends on the precise timing of signals between neurons. In some cases, the firing of one neuron must occur within milliseconds of another for the circuit to work correctly. This synchronization is achieved through both electrical and chemical communication, fine-tuned by countless feedback loops. Organs, made of multiple tissues, take coordination to yet another level. The stomach, for instance, is not just a bag for holding food. It is a complex organ composed of muscle tissue for churning, glandular tissue for secreting digestive enzymes and acids, and epithelial tissue for lining and protecting its interior. These tissues must work together seamlessly. The muscles must contract at the right time. The glands must secrete in response to the presence of food, and the lining must maintain its integrity, despite the acidic environment. All of this requires an unending exchange of signals within and between the tissues. In some systems, coordination crosses the boundaries between separate organs. The digestive process is a good example. When you begin eating, sensors in your mouth and stomach send signals that trigger the release of hormones and nerve impulses affecting distant organs. The pancreas prepares digestive enzymes, the liver increases bile production, and the intestines adjust their movement. Each of these organs responds not only to local signals but also to the needs of the entire digestive system. The immune system offers another striking example. When a pathogen is detected in one part of the body, immune cells in that region release signals that mobilize distant immune cells and organs, such as the bone marrow, to produce more defensive cells. This global coordination ensures that the body's defenses are ready wherever they are needed. The secret to this harmony lies in the way signals are layered and integrated. Local communication between neighboring cells provides fine-grained control, ensuring that small adjustments can be made quickly. Longer-range signals, hormones, and nerve impulses provide overarching direction, aligning the activities of distant tissues. Feedback between these layers ensures that the entire system remains balanced and responsive. If we could observe the coordination within a tissue or organ, it might resemble a city at work. Each cell would be like a citizen with a specific role, some transporting goods, others producing materials, still others maintaining structures. Streets and pathways, representing the cell-to-tissue connections, would be alive with the movement of messages and materials. Central signals would be like the city's planning office, guiding overall direction, while local conversations would keep each neighborhood functioning smoothly. Even as you sit in stillness, your tissues and organs are in constant motion beneath the surface. Your lungs expand and contract in rhythm, their airways adjusting diameter in coordination with the demand for oxygen. Your liver quietly balances the flow of nutrients, deciding which to store and which to release. Your skin replaces aging cells with new ones, knitting them into the barrier that shields you from the outside world. Each of these processes depends on countless cells working together in ways that no single cell could manage alone. This unity of action is one of the most beautiful features of life. It is what allows a body made of trillions of individual parts to function as one being. Without coordination, tissues would act independently, pulling in different directions, and the whole would lose its balance. With coordination, the body becomes a living symphony, each section distinct, yet all playing in harmony toward the same quiet, ongoing purpose: to sustain life. 16. Communication in single-celled organisms. It can be tempting to think of communication as something that belongs only to complex, multicellular life, to bodies made of many specialized parts that must coordinate their work. But the truth is that communication is far older than this. Long before the first multicellular organisms appeared, the Earth's oceans, lakes, and soils were already alive with exchanges of information between single-celled beings. A single-celled organism may be only a fraction of the size of a grain of sand, yet it still needs to sense its environment, respond to changes, and sometimes cooperate with others. In the absence of a nervous system, these creatures rely entirely on chemical and physical signals to navigate their world. And though their forms are simple compared to animals and plants, their ways of sending and receiving messages are surprisingly sophisticated. One of the best-known examples of such communication is a process called quorum sensing. This is a way for bacteria to measure how many others of their kind are nearby. Each bacterium releases small signaling molecules into its surroundings. As the population grows, these molecules accumulate in the environment. When their concentration reaches a certain threshold, the bacteria detect them and change their behavior in unison. Through quorum sensing, bacteria can coordinate group activities that would be useless if done alone. For instance, some species wait until their numbers are high before releasing enzymes to break down food, ensuring that there are enough participants to make the effort worthwhile. Others use quorum sensing to decide when to form protective layers called biofilms, thin, slimy coatings that anchor them to surfaces and shield them from harm. Biofilms are themselves a form of communication-driven cooperation. Inside a biofilm, bacteria are embedded in a shared matrix of sugars and proteins, creating a stable microenvironment. Within this community, signals pass more easily between cells, allowing them to adjust to changes in nutrient levels, oxygen availability, or threats from antibiotics. Biofilms form on rocks and streams, on the roots of plants, and even on human teeth as dental plaque. Communication in single-celled organisms is not limited to members of the same species. Some bacteria can send signals that influence entirely different kinds of organisms. In the soil, for example, bacteria may release molecules that attract the roots of plants, beginning a mutually beneficial partnership. Certain marine microorganisms use chemical cues to attract or repel tiny animal larvae, influencing where those animals settle. Even single-celled eukaryotes organisms like amoebas, algae, and yeasts rely on communication to thrive. In the freshwater amoeba Dictyostelium, communication takes an extraordinary form during times of scarcity. When food becomes scarce, individual amoebas begin to release a chemical signal that draws others toward them. The amoebas gather into a mound, fuse into a multicellular structure, and form a spore-producing body that can disperse to new environments. This remarkable transformation is entirely dependent on the chemical messages that draw the individuals together. Yeast cells, too, communicate with one another. During reproduction, yeast of different mating types release specific signaling molecules called pheromones. These pheromones are detected by receptors on potential partners, prompting them to grow toward one another and fuse. In this way, chemical communication ensures that reproduction occurs only between compatible cells. Some single-celled algae can sense and respond to light through specialized receptors. These photoreceptors allow them to detect the direction and intensity of light, guiding their movement toward optimal conditions for photosynthesis. While this is often thought of as a sensory response rather than communication, the distinction blurs when light-driven changes in behavior influence the position and distribution of entire populations, effectively altering the group's collective interaction with the environment. For many microorganisms, communication is also a matter of survival in the presence of threats. When bacteria are attacked by viruses called bacteriophages, they may release distress signals that trigger protective changes in nearby cells. Some may alter their surface proteins to avoid detection, while others enter a dormant state until the danger passes. This rapid, coordinated change increases the odds that at least part of the population will survive. In aquatic environments, planktonic microorganisms often engage in dense and complex communication networks. Chemical signals can indicate the presence of predators, the availability of nutrients, or even the approach of seasonal changes. These cues may influence whether the organisms remain free-floating or clump together in colonies, alter their buoyancy to change depth, or adjust their reproductive timing. The exchange of information among microorganisms is so fundamental that some scientists describe it as a kind of microbial language. This language is spoken in molecules, not words, but it is rich in meaning. Different molecules can convey urgency, invitation, warning, or cooperation. In some cases, the same molecule may have different meanings depending on its concentration, much like how the tone of a voice can change the meaning of a sentence. If we could observe this microscopic conversation, we might see a shifting cloud of invisible messages moving through water, soil, or tissue. Some signals would be short-lived bursts, fading quickly after they are released. Others would persist, slowly building in strength until they reach a tipping point that triggers a sudden change in the group's behavior. And everywhere, tiny cells would be responding, altering their movement, metabolism, or structure in response to what they have heard. What makes communication among single-celled organisms especially striking is that it is not merely reactive. Many microorganisms seem to anticipate future conditions by reading patterns in the signals they detect. In a way, they are making predictions. If the signal is rising quickly, it may mean that resources are becoming scarce, or that a cooperative opportunity is approaching. Acting on these predictions can mean the difference between survival and extinction. Communication in single-celled life also reminds us of an important truth. Cooperation and coordination are not inventions of complex life, but deep-rooted strategies that have existed for billions of years. The same principles that allow trillions of human cells to function together in harmony, sending messages, integrating information, acting collectively, can be found in communities made up of independent, microscopic individuals. Even now, all around us and within us, single-celled organisms are engaged in their quiet conversations. In the soil beneath our feet, bacterial signals are guiding the growth of plant roots. In the oceans, plankton are adjusting their behavior in response to the chemical traces of distant neighbors. Inside the human body, trillions of bacteria in the gut are not only talking to each other, but also influencing the activity of the cells, lining our intestines, and even sending messages that affect the nervous system. To study these simple yet profound exchanges is to see the deep continuity of life's communication systems. From the smallest bacterium to the most complex organism, life endures through the ability to share information, to act together, and to respond to change. In this way, the chatter of single-celled organisms is not a primitive precursor to real communication. It is communication in its purest, oldest form, the original language of life, still spoken in every corner of the living world. 17. Communication between different organisms. Communication in life is not limited to exchanges between cells of the same type, or even between cells within the same organism. Across the living world, signals often pass between entirely different species, linking their lives in ways that can be subtle, fleeting, or profoundly long-lasting. This cross-species communication shapes ecosystems, guides cooperation, and sometimes sparks competition. It is an ancient and essential thread in the web of life. One of the most familiar forms of this communication is the relationship between flowering plants and their pollinators. Flowers do not speak in sounds, but in colors, patterns, and scents. The bright petals of a daisy, the intricate markings on an orchid, the deep perfume of a jasmine bloom, each is a signal to visiting insects, birds, or bats, that nectar is available. The pollinator, in turn, responds to this invitation by moving from flower to flower, carrying pollen with it and ensuring the plant's reproduction. This exchange is mutual. The pollinator gains nourishment, and the plant achieves fertilization. Plants send signals not only to attract partners but also to deter threats. Many release volatile chemicals when bitten by herbivores. These airborne signals can be detected by nearby plants of the same or even different species, prompting them to strengthen their defenses before they too are attacked. Some plants go further, producing scents that attract predators or parasites of the herbivore, effectively calling for reinforcements from another species. In the oceans, corals engage in a quieter but no less intricate form of cross-species communication with microscopic algae that live within their tissues. The algae perform photosynthesis, producing sugars that feed the coral, while the coral provides the algae with shelter and nutrients. Signals pass between them to regulate this delicate balance. When conditions are stressful, perhaps water temperatures rise, the communication can break down, leading to the loss of the algae and the coral's bleaching. Beneath our feet, in the hidden world of soil, plants and fungi form vast underground communication networks. Many plants form partnerships with mycorrhizal fungi, which extend thread-like filaments into the soil to gather water and nutrients. In exchange, the fungi receive sugars from the plant. Signals between plant roots and fungal cells guide the growth of these networks and regulate the exchange. Some scientists have described this as a kind of wood wide web, where information about nutrient availability, pest attack, or environmental stress can travel from one plant to another through fungal connections. Animals, too, rely on cross-species signals for survival. Birds may follow the calls of other bird species to find sources of food or to detect predators early. Certain species of fish school together with unrelated species, using each other's movements as cues to evade predators more effectively. In the African savanna, some antelope species pay close attention to the alarm calls of monkeys, while the monkeys respond in turn to the antelope's sudden flight. Some relationships are more intimate still, shaped by long-term coevolution. Lichens, for example, are not single organisms but partnerships between fungi and algae or cyanobacteria. The fungus provides structure and protection, while the photosynthetic partner produces food. The signals between these partners coordinate growth and response to environmental conditions, allowing lichens to thrive in some of the harshest habitats on Earth. In the microbial world, cross-species communication is common and influential. In the human gut, for instance, trillions of bacteria interact not only with each other, but also with the cells of the host's digestive system. Some bacterial signals encourage the production of mucus that lines the gut, while others influence the immune system's readiness. Recent research suggests that bacterial signals can even affect the nervous system, influencing mood, appetite, and behavior, a reminder that cross-species communication can reach into the most personal aspects of life. There are also cases where cross-species communication involves deception. Certain orchids produce flowers that mimic the shape, color, and scent of female insects. Male insects, mistaking the flower for a potential mate, attempt to interact with it, and in the process pick up pollen. The insect leaves with no reward, but the orchid achieves pollination. Some predators use similar trickery, mimicking harmless species calls to lure prey into range. Marine environments offer their own forms of intricate signaling. Cleaner fish, for example, advertise their services to larger fish by performing distinctive dances and displaying bright colors. The larger fish approach and allow the cleaner to remove parasites from their skin, gills, and mouths. Both species benefit, and the interaction is made possible by clear, reliable signals that prevent the larger fish from mistaking the cleaner for food. Cross-species communication can also operate across vast distances and timescales. Migratory birds respond to seasonal changes in plant growth, following the waves of blooming and fruiting as they move along their routes. In turn, these birds disperse seeds, shaping the plant communities they visit. Whales and seabirds may follow schools of fish that themselves respond to changes in ocean currents and plankton blooms, an intricate chain of responses, linking organisms from the smallest to the largest. Even the simplest of signals can have profound effects when they pass between species. In some cases, these signals create entirely new opportunities for survival. In others, they form the basis of ancient relationships that have persisted for millions of years. The reliability of these exchanges is often built on repeated interactions, where both species learn in the broad sense of the word to trust the signal and respond appropriately. If we could step back far enough to see these interactions unfolding together, the living world would appear as a tapestry of exchanges. Colors, scents, sounds, movements, and chemical traces would weave through forests, grasslands, reefs, and rivers, linking species into an interconnected whole. The calls of birds would ripple through the canopy, chemical plumes would drift through the soil, light patterns would flash beneath the waves. Every signal would connect at least two lives, and often many more. This exchange across species boundaries is one of the quiet forces that hold ecosystems together. It allows different forms of life to fit together into stable patterns, sharing resources, protecting each other, and sometimes competing in ways that keep populations in balance. Without these signals, many relationships would break down, and the intricate web of life would fray. Even now, as you read, cross-species communication is shaping the world around you. In gardens and forests, pollinators are following floral cues. In the oceans, coral reefs are adjusting their partnerships with algae. In the soil beneath your feet, plants and fungi are trading messages and resources. These conversations are as much a part of life's story as the beating of a heart or the growth of a leaf, continuous, essential, and endlessly varied. 18. When communication breaks down. The story of life's communication is usually one of harmony, countless signals passing between cells, tissues, and species to keep systems balanced and alive. But like any form of communication, these exchanges can falter. Sometimes the message never arrives. Sometimes it arrives, but is misinterpreted. And sometimes the message is sent when it is not needed, overwhelming the system with noise. In the living world, the breakdown of communication can lead to misunderstanding, imbalance, and in some cases, the loss of life itself. Within the body, one of the most common points of failure occurs when receptors, the cell's listening structures, no longer recognize or respond to their signals. This can happen if a receptor's shape changes due to mutation, damage, or chemical interference. Without the ability to detect its messenger, the cell is left unaware of important instructions. In diabetes, for instance, cells may fail to respond properly to insulin. The hormone is present and the message is sent, but the receptor's response is weakened or absent. As a result, glucose builds up in the blood instead of being absorbed into cells for energy. Signals can also be lost if the pathways that carry them are interrupted. Damage to nerves can block the transmission of electrical impulses, cutting off communication between the brain and the body. This is what happens in certain injuries to the spinal cord, where regions of the body below the injury lose voluntary control and sensation because the messages from the brain can no longer reach them. In other cases, communication falters not because the message is absent, but because it is too strong. The immune system offers many examples of this. In autoimmune disorders, cells misinterpret normal, healthy signals as signs of danger. The immune system launches an attack against the body's own tissues, as if defending against an invader that is not there. The signals in such cases are clear, but they are wrongly interpreted, turning the body's defenses inward. Cancer, too, can be seen as a breakdown in communication. Normally, Cells receive and obey signals telling them when to divide, when to repair damage, and when to stop growing. In cancer, cells stop listening to these controls. They may ignore stop signals, continue dividing when they should rest, or even produce their own go signals to fuel their growth. The breakdown is not just in the reception of messages, but in the entire regulatory conversation that keeps cell populations balanced. Some failures of communication come from outside interference. Viruses, for example, can hijack a cell's signaling machinery, sending false instructions that benefit the virus at the cell's expense. Certain toxins disrupt nerve signaling by blocking the release or reception of neurotransmitters, effectively silencing vital messages between nerve cells. Environmental pollutants can also mimic or block hormones, distorting the body's endocrine communication and leading to developmental or metabolic problems. In ecosystems, communication breakdowns can have equally profound effects. If pollinators disappear from an environment, the plants that depend on them lose their means of reproduction. The flowers may still produce their visual and chemical signals, but with no receivers to interpret them, those messages fade into silence. Similarly, if a species that normally warns others of predators is removed, the web of alarm signals that once rippled through the community is weakened, leaving many animals more vulnerable. Climate change is introducing new forms of disruption. Seasonal cues that once guided migration, flowering, and reproduction are shifting. Birds may arrive at breeding grounds before their food sources have appeared. Plants may bloom before pollinators emerge. The signals themselves, daylight length, temperature changes still occur, but their timing no longer matches the conditions they once reliably predicted. The communication between species that depended on these shared cues becomes strained or breaks entirely. Even microbial communities, which have relied on stable chemical signals for billions of years, can suffer from broken communication. Antibiotics, while essential for treating infections, can disrupt the signaling networks within beneficial bacteria, altering their balance and reducing their ability to cooperate. In the gut, such disruptions can impair digestion, weaken immune regulation, and allow harmful species to take over. The consequences of communication breakdown vary. Sometimes they are small, a delay in response, a slight inefficiency. Other times they are profound, cascading through systems until entire functions are lost. In the heart, for example, misfiring electrical signals can disrupt the rhythm of contraction, reducing the heart's ability to pump blood effectively. In the brain, disrupted neurotransmitter signaling can lead to disorders of movement, mood, or cognition. If we could see a breakdown in communication as it happens inside a cell, we might notice that the familiar patterns of interaction are missing. The receptor waits, but the signal never arrives. Or the signal is present, but the pathways downstream remain silent. In another corner of the cell, messages race ahead without restraint, overwhelming the normal checks and balances. The harmony of movement and response gives way to either stillness or uncontrolled activity. Life is resilient, and many systems have built-in safeguards to limit the damage when communication falters. Redundancy is one such safeguard. Multiple pathways can sometimes carry the same message, so if one fails, another may take over. Feedback loops can dampen excessive signals before they cause harm. In ecosystems, diverse species can sometimes fill similar roles, maintaining key functions even when one species' signals are lost. Still, there are limits to this resilience. Communication is the invisible thread that holds life together, and when too many of those threads are cut, the pattern begins to unravel. The breakdown is often gradual, a slow erosion of connections that may go unnoticed until the consequences are undeniable. By the time flowers bloom without pollinators, or nerve impulses fade in damaged tissue, the silence is already deep. Understanding these breakdowns is not only important for medicine, ecology, and conservation, It is also a reminder of the quiet complexity that sustains us. Every heartbeat, every step, every exchange between species depends on signals moving faithfully from sender to receiver. To keep those signals clear is to protect the very fabric of life's ongoing conversation. 19. The resilience of life's communication systems. In the vast and intricate network of life, communication is both delicate and robust. It can falter under strain, as we have seen, but it also possesses a remarkable ability to recover, adapt, and continue. This resilience is one of life's defining traits, the quiet strength that allows signals to find new paths, conversations to resume, and harmony to be restored after disruption. At the cellular level, resilience often begins with redundancy. Many essential messages can be carried by more than one pathway. If one route is blocked, another can often take its place. In nerve cells, for example, multiple types of ion channels can contribute to the transmission of electrical signals. If one type is damaged, the others can help maintain communication, even if at reduced efficiency. Similarly, in hormonal signaling, overlapping systems ensure that critical processes like growth, reproduction, and metabolism are not wholly dependent on a single molecule or receptor. Cells also possess repair mechanisms to restore damaged components of their signaling machinery. Proteins, receptors, and ion channels that have been altered or degraded can be broken down and replaced. DNA damage that affects the instructions for these proteins can sometimes be repaired directly, allowing the cell to resume normal function. These quiet acts of molecular maintenance happen constantly, ensuring that the cell's ability to listen and speak remains intact. Adaptation is another cornerstone of resilience. Cells can adjust their sensitivity to match the availability and importance of a signal. When receptors are lost or damaged, cells may produce more of them to improve their chances of catching the message. If a signal is consistently strong, the cell may reduce its sensitivity to prevent overstimulation. This flexibility allows communication to remain balanced even in changing or challenging conditions. In tissues and organs, resilience emerges from cooperation. Neighboring cells can share resources, reinforce each other's signals, and take over some of each other's roles when necessary. In the heart, for example, if a group of pacemaker cells fails, others can step in to maintain the heartbeat. In the liver, healthy cells can increase their activity to compensate for the loss of function in damaged areas. This cooperative adaptability helps preserve the performance of the whole, even when individual parts are struggling. The nervous system shows remarkable examples of communication resilience. After injury, neurons can sometimes form new connections, rerouting signals around damaged areas. This process, called neuroplasticity, allows the brain and spinal cord to reorganize their networks, recovering lost functions or developing new ways to perform them. While such adaptations may not fully restore the original pathways, they often allow the system to regain much of its capability. In the immune system, resilience is expressed in its ability to learn and remember. When pathogens disrupt normal communication between immune cells, the system adapts by refining its signals for the next encounter. Memory cells store information about the invader, ensuring that if it returns, the response will be faster and more effective. This memory is a form of lasting adaptation, a reinforcement of the immune system's internal conversations. Resilience is not confined to the body of a single organism. In ecosystems, communication networks between species can reform after disruption. When a pollinator species declines, other pollinators may expand their range or change their behavior to fill the gap. Plants can sometimes shift their flowering times or produce stronger visual and scent cues to attract new partners. Predator-prey relationships can adjust as well. with new warning signals or escape strategies, evolving over time. Microbial communities, too, display resilience in their signaling. When antibiotics disrupt the balance of bacteria in the gut, surviving species can adjust their communication patterns to stabilize the community. New bacterial arrivals from food, the environment, or probiotic sources can join the network, restoring lost functions. This adaptability helps maintain essential interactions between microbes and their host. Some of life's resilience comes from its ability to create entirely new channels of communication when old ones fail. In certain plants, when traditional chemical signals to resist pests are disrupted, they can produce alternative compounds that serve the same purpose. In animals, when vocal signals are masked by noise, visual or tactile signals may take on a greater role. Even at the cellular level, new signaling pathways can evolve to meet changing needs, adding to the network's diversity and flexibility. Resilience often works quietly. Over time, signals that were once faint can grow stronger through repeated use. Weak pathways can be reinforced through structural changes, as in the strengthening of neural connections through learning and practice. The result is not always a return to the exact state that existed before disruption, but a new stability, one adapted to the present conditions. If we could observe resilience as it unfolds, it might appear as a patient rebuilding process. Damaged pathways would slowly regain their flow. Alternative routes would emerge, weaving around obstacles. The gaps left by lost signals would be bridged by new ones, sometimes different in form but similar in function. Over time, the network would regain its coherence, like a web mended by the careful work of its builder. This capacity for recovery is not limitless. Some disruptions overwhelm the system's ability to adapt, leading to lasting loss. But the very fact that life so often finds ways to restore communication is a testament to its deep commitment to connection. Communication is not simply a convenience for living things, it is a necessity, woven into the very fabric of survival. And because it is so essential, life has evolved countless strategies to preserve it against interruption. Even now, in your own body, resilience is at work. Cells are repairing damaged proteins. Neural pathways are adjusting their connections. Hormonal systems are fine-tuning their balance after small daily stresses. Around you, ecosystems are reshaping their relationships, adjusting the flow of signals between species to match new realities. Much of this happens unnoticed, yet it is the quiet work that allows life to persist through change. Resilience in communication is not about perfection. It is about persistence, the ability to continue the conversation, even if the words must change. It is about finding new routes when the old ones are blocked, about listening more closely when the signal is faint, and about never letting silence fall for long. It is this persistence that has allowed life to endure through billions of years of shifting climates, changing landscapes, and constant challenges. In the end, resilience is the silent partner of every message that passes through the living world. It waits in the background, ready to restore the thread when it is broken. And because of it, the conversation of life continues unbroken across generations, steady even through the storms, always finding its way back to harmony. 20. The harmony of life's conversations. From the smallest bacterium to the most complex living organism, life is bound together by conversation. Every signal, whether a fleeting pulse of electricity, a drifting molecule, a burst of light, or a quiet chemical whisper, is a part of this ongoing exchange. Over billions of years, these conversations have grown in richness and variety, weaving connections within cells, between cells, across tissues, through organs, and even between entirely different species. The result is a vast, intricate web of communication, as constant as the rhythm of the tides, and as essential as breath. We have seen how cells speak in many voices, through direct contact, by releasing signals to their neighbors, by sending hormones across the body's circulatory highways, and by passing quick chemical messages at synapses. We have seen how they amplify small signals into great ones, and how they fine-tune their responses with care and precision. We have followed messages into the depths of the cell, where second messengers carry them to their destinations, and watched as cells combine multiple inputs into a single, balanced decision. We have also explored communication beyond the boundaries of a single organism, between pollinators and flowers, between corals and algae, between fungi and the roots of trees. In the soil, in the oceans, in the air, messages pass invisibly from one form of life to another, guiding cooperation, warning of danger, and shaping the flow of energy through ecosystems. Even the smallest independent organisms, from bacteria to single-celled algae, share in this ancient tradition, releasing signals to guide their movements, their growth, and their relationships with others. The harmony of life's communication lies in its balance between diversity and unity. The signals are many and varied: electrical, chemical, mechanical, visual, and beyond, yet they all serve the same fundamental purpose: to connect living things to one another and to their surroundings. No single form of communication can meet every need. Instead, life relies on an interwoven network of different methods, each complementing and reinforcing the others. It is this balance that gives life its resilience. When one pathway is blocked, another may carry the message. When a signal is faint, it can be amplified. When it is too strong, it can be tempered. Across scales, from the whisper of calcium ions in a single cell to the migration calls of birds heard across continents, communication is flexible enough to adapt, yet precise enough to maintain order. There is also a kind of elegance in the way these conversations scale upward. A message received at the surface of a cell may lead to changes deep in its interior. Those changes, repeated across millions of cells, create a tissue-level effect. Tissues, working in concert, produce the coordinated actions of organs. Organs, connected through nervous and hormonal pathways, form the living whole. And that whole, in turn, sends and receives signals within its environment, shaping and being shaped by the other lives it touches. If we could step back far enough to hear all these conversations at once, it might sound like a symphony, not of melodies and harmonies in the human sense, but of patterns, rhythms, and flows. The rapid staccato of nerve impulses would blend with the slower, sustained tones of hormonal signals. The subtle background hum of local chemical messengers would mingle with the occasional bright flourish of amplified responses. Every note would have its place, every phrase its meaning, every pause its quiet importance. Yet, for all its complexity, this great conversation is not deliberate in the way human speech is. It is not planned word by word, but it is no less meaningful. Each message is shaped by need and by history, refined through countless generations of trial and error. It is communication in its most essential form, not to persuade or to entertain, but to survive, to grow, to sustain balance. The harmony of life's conversations is also a harmony of time. Some messages last only fractions of a second, guiding an immediate reaction. Others unfold slowly, over days, months, or even years, shaping growth, reproduction, and seasonal rhythms. And still others endure across generations, encoded not in fleeting molecules, but in inherited patterns of behavior and molecular pathways, lessons written into the very structure of life. In the human body, this harmony is present in every moment. Even as you rest, countless conversations are taking place inside you. Your heart cells pass electrical signals to keep their beat steady. Your brain cells exchange neurotransmitters to maintain awareness and coordinate thought. Your immune cells trade chemical cues that balance vigilance with restraint. In your gut, microbes send signals that influence your digestion and even your mood. All of it happens quietly, without conscious effort, as part of the constant background music of life. In nature, the harmony is no less constant. The opening of a flower sends a cue to its pollinators. The fall of a leaf feeds soil organisms, whose signals influence the growth of roots. The return of migratory animals signals the change of seasons to the ecosystems they visit. These exchanges happen everywhere, in every moment, from the surface of the sea to the deepest soil, linking all living things into a single, ongoing dialogue. And yet, this harmony is not static. It shifts and adapts, just as the melodies of a living song might change with each performance. New species enter the conversation, others fall silent. Environmental changes alter the timing and strength of signals. Communication systems evolve, finding new ways to connect and new balances to maintain. The conversation of life is always the same in its essence, yet always new in its details. To reflect on all this is to see communication not just as a feature of life, but as one of its most defining qualities. It is the means by which life organizes itself, by which it senses and responds, by which it maintains the fragile balance between order and change. Without communication, the intricate choreography of life would fall apart. With it, life can endure, adapt, and flourish across the vast sweep of time. As you close your eyes and let your breathing slow, you can imagine this conversation continuing a gentle chorus of signals moving through the living world. It is a song without a single composer, yet one in which every participant plays a part. It is a language without words, yet rich in meaning. And it is a harmony that, so long as life persists, will never truly end.